Good morning and good evening, everyone. Um, I welcome you all to our 20th Cancer Research Symposium, and this is the Cancer Research session. And we have four speakers with us, and I'm Saira Salim, and I work as a research scientist at the Basic Science Research Department. And today, our first speaker is Professor Rodney Scott, and he is a professor at the School of Biomedical Science and Pharmacy, Newcastle University, Australia. And his main interest is in the genetics of colorectal cancer and breast cancer. And he has published more than 500 scientific articles. We look forward to his talk, which is summarized as gene panel testing for breast cancer. Do we have enough information? Over to Professor Rodney.
Before I start this talk, I'd like to first of all thank the organizers, Professor Mohammed Asad Parvez, Professor Kashif Sayad, and Professor Mohammed Usman Rashid for inviting me to give this talk today at the Shaukat Khanum Cancer Symposium. I trust that the symposium is going very well and that it will be very informative for everybody. So without much further ado, I'll get into the talk. So today I'm just going to talk about gene panel testing for breast cancer. Have we got enough information? And the reason I want to put this title in is because there have been a number of genes that have been identified in recent years that may or may not be associated with breast cancer. Just because a gene is identified that carries a pathogenic variant or a variant that disrupts completely the coding sequence of that particular gene does not necessarily mean it's a breast cancer susceptibility gene. And it could well be just a gene that's been carried over into a breast cancer population that hasn't really been studied well enough in a control population to address those issues. We've known about new genes for quite some time, and there's a list here that goes back to 2010, where a number of genes are listed as high penetrance rare cancer predisposition genes. These include BRCA1 and BRCA2. There's also moderate risk cancer alleles, and these include genes like ATM, CHEK2, and PALB2, and genes that have been identified through genome-wide association studies, and they have low penetrance and high frequency in the general population. And the ones we're particularly interested in are those new genes that are associated with a high degree of penetrance and or those that are linked into moderate degrees of disease penetrance. So we're going to be talking briefly about BRCA1 and BRCA2. Then we're going to talk about how many BRCA genes are actually actionable and how valuable is this information, and what can you do to actually try and sift away those that are not so valuable against those that are very valuable. And towards the end of the talk will show you that we are now beginning to focus on the key players associated with breast cancer risk. So how do we go about identifying cancer predisposition genes? We can do two things. We can look at family studies, or we can look tumor pathology, or look at tumor pathology. Family history studies or family studies have been undertaken for many years now, and they've usually been linked into families where there's at least one case of early onset disease, or may well be a single woman with early onset disease, and usually under the age of 40 years. And there's been an assumption that these women are more likely to be associated with a genetic predisposition than those who develop breast cancer over this age. Another way of improving your hit rate is to identify multiple affected family members. And these include cases where there's one or more individuals under the age of 55 years of age. And there's also two remaining factors, those that are families that are associated with breast and ovarian cancer, and those that are linked with breast cancer and other malignancies. We also need to be aware that there are multiple different types of familial cancer syndromes. There's not only hereditary breast ovarian cancer, but there's also Li-Fraumeni syndrome, and there's a few others as well. However, family studies are now coming into question as there are calls to test all women with breast cancer for their genetic risk of disease. An alternative to looking at family studies is to look at the pathology of the tumours themselves, and is there anything special about them that may allow you to predict with a higher degree of certainty whether or not they carry a mutation or not. So because BRCA1 and BRCA2 have been looked at for many years, we know that BRCA1 mutations are more basal-like, they're more pleomorphic, they have a higher mitotic content, and medullary and atypical medullary carcinoma is more frequent. There's also less ductal carcinoma in situ in women who carry BRCA1 mutations. So these are all probabilities. They're not absolutely defined, but it's the probability of finding these features that's important. Similarly, with BRCA2 mutation positive tumors, there's less tubule formation. You have to be careful because there's no difference in pleomorphism or mitotic content between tumors arising on a background of BRCA2 mutations compared to those in the general population. However, one aspect that has clearly been identified over recent years is the fact that the triple negative phenotype, that is estrogen receptor, progesterone receptor, and HER2 receptors are negative, and these are significantly overrepresented in the BRCA1 and BRCA2 populations. So just to give you an idea, we looked at a study of around 800 individuals from Poland and Australia to determine whether or not triple negative breast cancer, or what relative percentage of triple negative breast cancer cases could be accounted for by BRCA1 and BRCA2. So we identified in our cohort around 10% of the total cohort had pathogenic variants identified in BRCA1 and BRCA2. 36 of the patients had a family history of disease, and 38 of them had no family of disease. The mean age of disease onset for the combined cohort was 52 years of age. And for non-carriers, the BRCA mutation cohort was 52 years, and for the non-carriers, it was 58.7 years. The BRCA1 mutation carriers had an average age of disease onset of 47.2 years of age, whereas the BRCA2 carriers had an average age of onset of 58.8 years of age. There were no differences in the average age of disease diagnosis between BRCA2 carriers and non-carriers, and that's shown in the next couple of slides. So this shows you the distribution of the age ranges of which you expect to see the most cases. So for BRCA1, it's somewhere between 35 and 45 years of age. For BRCA2, it's somewhere between 55 and 64 years of age. If you compare this distribution of the mutation-negative population, which is shown here, to that previous graph, you find that the BRCA2 mutation group mirrors that of what is observed in the non-carrier population that we studied. This is important information because it's actually suggesting that age of diagnosis, certainly for BRCA2 carriers, is not a very good indicator of whether or not you're going to carry a mutation or not. So the clinical implications of these findings indicate that about 10% of women with triple negative breast cancer have a BRCA1 or BRCA2 mutation. Patients with triple negative breast cancer should be considered as candidates for genetic screening. We should not restrict screening to women without a family history of disease, because 38 out of the 74 were identified as not having a family history of disease. Age restrictions should be relaxed, especially if you consider the age distribution of BRCA2 mutation carriers compared to the mutation-negative carriers. And out of this, we know that 10% of them at least will receive a better targeted therapy if they're offered BRCA1 and BRCA2 testing. Now, that goes hand in hand with the next question that we need to ask. How many breast cancer genes are actionable? So if we look at mutation detection, we find that firstly, we're able to do more for much less. The next generation DNA sequencing has revolutionized mutation detection. We can test more genes at less cost and decrease turnaround times than ever before. Commercial entities are driving genetic testing by offering panels that have ever increasing numbers of genes on there, because there's a belief that the more you have on in your panel, the more you're going to capture. That's not untrue, but it may not help in the context of identifying women with a genetic predisposition to breast cancer. The availability of gene panels include a wide variety of breast cancer susceptibility genes. But the question that we have to ask is how many of them are actually clinically actionable. Now, the risk genes are defined by the loss of function variants. Many of these genes have had no population control data assessed, and most of the genes that have been identified don't have disease penetrance estimates. Therefore, it's difficult to assign causality. So what we need to identify whether or not a gene can be used for clinical purposes is that the transmission of the phenotype is obvious, often at a younger age than normal ages. It's a very high level of disease penetrance, or high to very high, with it being over 50%. The mutations themselves are unequivocally pathogenic. The mutations are absent or present at very low rates in the general population or control population. And we need to be safe in knowing what the population carrier frequencies of these variants are likely to be. So with that in mind, we sort of looked at this list of genes here: ATN, ATR, BARD1, BRIT1, BLM, CHEK2, CDH1, MRE11A, MBN, RECQL, NF1, P10, RAD50, STK11, and XRC2. So the problem is with all of these genes, there's little or no information about disease penetrance. So we know nothing about age-dependent risk. There are very small numbers of patients identified to date with causative mutations. There's little, if any, information about diseases that are associated with these causative mutations outside of the context of breast cancer. There's no or little information about the presence of genetic variants in a healthy population. And there's little or no information about environmental risk factors that may promote disease development in women who carry these variants in these genes. So how do we go about addressing this shortfall in knowledge? Well, you could examine the frequency of mutations in a case control population. You can try to define disease penetrance. You can determine if mutations in specific genes alter the risk of a specific malignancy. And you can assess whether single genes are associated with multiple tumor types, such as pancreatic cancer and breast cancer, or prostate cancer and breast cancer. Or again, if you really are interested, you could look at ovarian cancer and breast cancer. And there's reasons for doing that. So what did we do? To try and address these shortfalls in knowledge, the first thing we did was look at population studies. We had the ability to identify 2,000 index patients in whom over 95% of them had breast cancer and less than 5% ovarian cancer. All were pre-screened for BRCA1 and BRCA2 mutations. So we knew that they did not have any variants in those genes, or the probability of identifying individuals with BRCA1 and 2 or 2 mutations is going to be very low. All patients in our index population were young, they were under 50 years of age, or they had a very strong family history of disease. Patients were collected between 1997 and 2014 and compared against 1,997 population controls who were cancer-free as of January 1st, 2015, and their average age was almost 60 years of age. And this was an institutional review board approved study. So what did we observe? Out of the total number of samples that were sequenced, which was close to 4,000, 94% of them, 94% of the coding regions of all genes were covered. We did identify 6 actionable mutations in BRCA1 and BRCA2. And those carriers were not included any further in the analysis. Out of the 1,994 patients who were left over, we identified 69 variants in the cases, and in the 1,985 controls, we identified 26 pathogenic variants. PALB2 contributed 22 pathogenic variants in the patient group and 3 in the control population. 5 patients harbored pathogenic variants in TP53, and none of them had a family history of disease. We also revealed that breast cancer alone was not a criteria to select for P10 or STK11 mutational testing. The pathogenic variant detection rate was stratified when we stratified it by age did not differ between the two groups. The presence of a significant number of pathogenic variants in the control group led to a positive predictive value of 0.73, and the population attributable risk was found to be about 2.1%, and just over half of the risk, population attributable risk, was due to PALB2. Aside from TP53 and PALB2, the contribution of the remaining 12 genes was, to say the least, modest. 2.1% of the patients harbored a loss of functional change, so that was represented in 42 individuals, and in the control population, 1.15% or 23 individuals had a functional change in one of these genes. And this was consistent with a modest risk ratio of 1.83. Now, that at the time was revealing and indicated to us that only some of these genes that we have identified should be used for genetic testing, because the majority of them didn't really reveal very strong associations whatsoever, especially in light of the fact that many of them were identified in a control population. More recent data that came out earlier this year, was published in the New England Journal of Medicine in February, and the questions that the BCAT group, which published this result, were asking was what genes are required for breast cancer genetic testing, how many genes still do not have sufficient information to warrant the use of an NGS panel NGS panel testing, what were the risk estimates like, and was there a breast cancer subtype specific risk associated with any of these variants. So the results that we see here indicate in the red boxes the sort of genes we might expect, BRCA1 and BRCA2, and this diagram shows only those nonsense mutations. So we have ATM, BRCA1, BRCA2, CHEK2, and PALB2 coming up with the highest odds ratios of risk, followed by BARD1, BRAD51C, 51D, and TP53. Had lower risk, but they were consistent. When we looked at the missense variants, the rare ones, we found that CHEK2 and TP53 carried the majority of them, and this was associated with increased risk of disease. So one needs to be careful what one looks at with respect to missense variants and how they're interpreted. If we look at this bar diagram, we identified on the right-hand side of the diagram, over here, the relative contributions of BRCA1, BRCA2, PALB2, TP53, and CHEK2, P10 and ATM to breast cancer risk. And we find that women with breast cancer were very much overrepresented having mutations in these genes. As we go more towards the left of the diagram, we find that the frequencies of the mutations that were tested match those that were observed in the controls, similar to what we observed in our initial study. Now that's important information and shows you that some of these genes are unequivocally associated with breast cancer risk. The BCAT consortium took it a stage further. On the left-hand side of the graph, we see the same risk pattern up here. But if we split it then into ER-positive breast cancer and ER-negative breast cancer, we find that the BRCA1 and BRCA2 and PALB2 risks are reduced somewhat compared to the ER-negative breast cancers, where, for example, BRCA1 has an overall risk of about 55 times that compared to the general population, whereas for ER-positive breast cancer, it's only about 5 times. Similarly, we find that BRCA2, PALB2, TP53 have higher rates of, or associated with a higher overall risk of breast cancer for ER-negative disease compared to ER-positive disease. Intriguingly, CHEK2, P10, and ATM are associated more with ER-positive disease than negative disease. Then, if you look further down at the ER-negative diseases, we see that BARD1, MSH6, RAD51C, and RAD51D, as well as potentially NF1, are associated with an increased risk of ER-negative disease, whereas it's not really associated with any ER-positive disease risk associations. Intriguingly, MLH1, AKT1, MEN1, and PIK3CA are associated or appear to be associated with protection from development of breast cancer. But all the genes in between, listed in here, are all hovering around population risk levels. And this indicates that the genes that have been identified to date that are unequivocally associated with breast cancer risk are associated with, for the main part, homologous recombination. And that's shown on the right-hand side of the graph, going from TP53 all the way up to BRCA1. And there's not an awful lot within this section of the graph that shows you that there's an awful lot more associated with ER-negative disease. It's important to remember, though, however, that BARD1, MSH6, RAD51C, and RAD51D, possibly NF1, are also associated with an increased risk of ER-negative breast cancer. So if you're going to include anything in a panel of genes that would be used specifically for breast cancer risk, it would be NF1, RAD51D, 51C, MSH6, BARD1, CHEK2, TP53, PALB2, BRCA2, and BRCA1. I would not recommend using P10 or ATM. However, if you look on over here for ER-negative disease, yes, you'd certainly want to include CHEK2, P10, and ATM. If we look at the penetrance, age-specific penetrance at 80 years of age, we find BRCA1 is by far the highest, with it being somewhere around 55% by 85 years of age. BRCA2 and PALB2 are somewhere between 40 and 45% risk at 80 years of age. CHEK2, BARD1, ATM, RAD51 and RAD51D decrease with risk down to around somewhere between 15 and 20%, and population risk is hovering at around 12%. And these give you a relative indication as to what the general risk of these genes are with respect to disease. But the question then arises, whom to test and whom not to test? Well, around 3% of breast cancer patients carry a pathogenic variant of BRCA1, BRCA2, and PALB2, and they have a greater than 4-fold risk of disease, and this would be risk as a high-risk, high-penetrant disease gene. 3% of the breast cancer patients carry a pathogenic variant in ATM, CHEK2, and they have a 2 to 3-fold increased risk of disease. So we call these an intermediate risk group. If you use the familial approach and you just focus on familial cases, you'll find that the greater pathogenic variant detection rate misses about 0.8% of all individuals. Comprehensive screening could be offered to all women, but it's expensive and it's potentially harmful. So you have to be very careful in how you interpret the data. And it's probably a better way to go would be to look at testing all women over the age of 65 years of age who have triple-negative breast cancer, where you're going to pick up about 3.3% of the total number of these women having a pathogenic variant, and therefore you actually want to test them. And you probably don't want to test those who are ER and PR positive. Having said that, any woman presenting with triple-negative breast cancer under the age of 65 years of age should automatically be tested anyway. So in summary, family history, even though it's sort of beginning to wane in popularity for the identification of women at risk, is still the simplest method to identify women who have high risk. Tumor pathology reveals about the same percentage of BRCA variant carriers compared to family history. new genes associated with breast cancer risk have been identified, and many of the new genes still require more supportive evidence with respect to causality. And one of the challenges there is that they are vanishingly rare, and it's going to take quite some time to accumulate sufficient information to assign causality to these genes, and that will probably only be achieved by massive collaborative studies. With that, I'll end this talk and take any questions. Thank you.
Thank you very much for your excellent talk, Professor Rodney. I would like to remind all our respectable audience that please type your questions in the chat window, and we'll take all the questions at the end of the session, at the end of all the 4 talks. If we could not discuss all the questions, we'll direct those questions to the relevant speakers. So with this excellent talk, I would like to introduce our next speaker, Professor Dr. Jörg Hoheisel. He heads the Division of Functional Genome Analysis at the German Cancer Research Center in Heidelberg, Germany. And his research objectives are the assessment and description of the regulation of cellular functions, with a particular focus on the analysis of protein variations and by means of affinity proteomics. Another line of work is a very interesting one, which is on synthetic biology for the establishment of artificial molecular systems. And we very much look forward to his talk, for which the title is the Proteomic Analysis in Tumor Biology, Mechanistic Insights, Personalized Diagnostics, and Novel Therapy Options. Over to Professor Dr. Jörg.
Hello, this is Jörg Hoheisel speaking. I'm working at the German Cancer Research Center in Heidelberg in Germany, and I'm going to talk about the use of some techniques of proteomics for the improvement of knowledge about pancreatic cancer, or cancer and pancreatic cancer in particular, and the kind of work we are pursuing to actually make that work. So I will share my screen with you to show the presentation I have. Here we go. We don't. Oh, yes, we do. So I should also tell you in the beginning that besides scientific interests, I also have some commercial interests due to a patent that we filed. And also, since I'm a co-founder of a company dealing in the antibody kind of work that I'm going to talk about also. Heidelberg is located in the southwest of Germany, so somewhere in the middle of Europe for that matter, and it's an old city, relatively speaking. So this is the old city center of Heidelberg. And 2 bridges down the river, it's the area of Heidelberg that I'm in most of the time. So this is the campus where most, or basically all the natural science faculties of the university are located. Also, nearly all university clinics are meanwhile located here, and several independent research institutions, including the German Cancer Research Center, which is a national research center on cancer research, as the name says, and has some 3,500 people working on all aspects of cancer research. I'm located in the green building at the top right. So in case you are going to visit me, this is where you can find me, on the 4th floor. My scientific interest is mostly pancreatic cancer, all kinds of aspects of pancreatic cancer, well, mostly molecular ones, of course. And one of the reasons is that pancreatic cancer is one of the few cancer diseases for which there is basically no treatment. About 20% of the patients can undergo surgery, so resection of the tumor. But even those people eventually mostly die of pancreatic cancer. This high mortality, which is basically identical to incidence, is the reason why pancreatic cancer is going to be the 2nd most deadly cancer worldwide, or let's say in Western Europe and North America mostly. Second only to lung cancer, while all the others are unfortunately being surpassed in terms of number of deaths of the respective disease. The reason for that is there's basically not much of treatment, and also pancreatic cancer has been diagnosed late, and that's the main reason why treatment doesn't do much good anymore. So we want to change this, and in order to actually achieve this kind of objective, or at least partly, what we are working on is a molecular analysis on all molecular levels you can think of, mostly DNA, RNA, and protein, but also on the serum or other secretome of cells, cell lines, the proteome of the cell lines themselves, and so on and so forth. So the idea is to gather a lot of molecular data and then use them in function. In this network, regulated by this gene again, was dopamine receptor D2, which was interesting, besides others, for the main reason that actually quite a few of inhibitors already exist and are in clinical use that inhibit DRD2. That was the main interest. We, of course, confirmed that this highly significant, but only slight increase at the mRNA level, as you see at the top left, is actually translated into quite a change at the protein level. If you take into account the location of the cells that exhibit high protein levels, is actually even stronger than that. So, which means DRD2, by a lot of experiments we did, was found to be really important for the development of pancreatic cancer. And as said, there are already several inhibitors, one of which is haloperidol, for instance. And they are in use to treat diseases but cancer. So for instance, schizophrenia and other mental health diseases are being treated with these DRD2 inhibitors. So which means they are in place, they are well characterized in terms of side effects they have and so on and so forth. And we looked at what effect they may have actually in, well, animal experiments. And this is shown on the right. What you see is for once, at the bottom 5 mice that had pancreatic cancer treated with haloperidol, at the top 5 controls. We have done many more than those 5 animals, but that's just showing a few examples. What you can see is the area, labeled in white is primary tumor. It does exist because it existed before we started the treatment, but the point is it didn't grow further, which means, it really stopped growing, which means upon treatment with haloperidol or pemoxide, actually primary tumor growth stopped and even shrunk a little bit. But more interesting, I think, and particularly in terms of a clinical application, is this mouse here was the only one where we found metastases in the mouse, and just a very little one, a very small one. While in all controls, and as I said, we did more than the few shown here, the yellow labeled area is indicating the metastases, and you can see all of them have many and big ones. As a matter of fact, what we found, but for this one mouse, none of the others after treatment developed metastasis, and those mice that had metastasis before we started the treatment, there we found a reduction in the size and volume of the metastasis, which means what we have at hand is actually a protein, of which we know that once haloperidol or another inhibitor is binding to it, that actually ER stress has been triggered, which we found out in a lot of experimental work, of course. Basically, or nearly all of the tumors show very high levels of DRD expression, which means inhibition could and should work. Also, the stem cells of pancreatic cancer show extremely high DRT2 levels, which means inhibitors also work there. We can avoid any possible resistance once we go into clinical application, because there are quite a few of different inhibitors of DRT2, so even that could be covered. And last but not least, there are other drugs that also affect the relevant pathways, like for instance, atorvastatin and others shown below here. So what you see is they even further the effect. So basically there's no further growth of tumor at all in this co-treatment kind of effect. So what we have at hand is now really drugs that are already in clinical use, and we are setting up a clinical trial, in-house clinical trial, where we are actually applying this to patients, and in, let's say, about 2 years, I should be able to tell you what this will do in real terms on patients of pancreatic cancer. What also, of course, is of interest is liquid biopsy, which is a way to hopefully find cancer early, because what you do is you take some blood samples. Urine is another option, but what we talk about is blood samples from people. You do it either for screening, which means finding people who have the disease. You do it for the purpose of early detection, which is also, if you like, could be connected to the screening, because as said, once pancreatic cancer would be detected early, the survival rate would be much higher. Could be, as said, usually it's detected very late indeed. And also what can be important, actually is important, is differential diagnosis, where we actually want to make sure that a person who has pancreatic cancer has cancer, and that's not another pancreatic disease like chronic pancreatitis. And actually the distinction between the two, one cancer, the other one just being an inflammation, which is bad, but not that bad, is 65%, which means in a third of the cases, the distinction initially is not possible. So there are false results either way, both, chronic pancreatitis patients being called pancreatic cancer and vice versa. So what we are doing is we actually work on various aspects, analyzing blood, one of which is, for instance, studying the free nucleic acid circulating and methylation as one means to distinguish cancer patients from other individuals. That is working very nicely, and I'm going to talk about this tomorrow in a bit more detail. Also, what we look at in terms of proteomics is actually circulating antibodies. So antibodies circulating in the blood of the patient that may be indicative of a tumor being around. And what you see here is actually what we do is we have spotted on surfaces, glass surfaces, some 3,000 proteins, mostly surface proteins, from the membrane or outside of the membrane, but also secreted proteins and others similar. Isolate, well, so far we have studied 1,200 sera from patients and control people who have, let's say, chronic pancreatitis or other pancreatic diseases or purely healthy. And what we are aiming at is really finding binding of antibodies from the sera to particular proteins which are reproducible and specific, of course, for people who have pancreatic cancer. That seems to work reasonably all right. As said, this analysis is still ongoing, so the idea is to complete some 1,500 patient sera and then be able to tell how well we are able to pick up tumor. As it looks, specificity is extremely high, but the number of similar signals between all these patients is relatively small, which means we have to pick different or define different signatures to hopefully then eventually finding out how to actually pick up at high specificity and sensitivity pancreatic cancer patients from normal and healthy individuals. What we also do is we look at protein circulating in the blood. The way we do this is that we actually attach antibodies on the glass surface, and not just one, obviously, but quite a few. And actually, again, about 3,000 antibodies, some 3,000 antibodies binding actually 2,600 proteins. So each spot here represents one particular antibody that is bound by the surface. Then what we do is we isolate the protein from the blood, label it with a fluorescence dye, red or green, healthy, let's say, and red is the tissue or the proteins from sera from pancreatic cancer patients, and incubate this on this antibody array, and the protein is binding, and by the color of the fluorophore attached to it, we are able to pick up which one is present and in what amount it is. That's a semi-quantitative, but working quite nicely. And showing just one example, what we've done is here is actually we compared the proteins that are present in the secretome of pancreatic cancer cells, of quite a few of them. So I think it was 24 different ones. So the molecules that are being, proteins that are being secreted by the tumor cells. And compared this to the proteins that we found in zero from PDAC patients, and you can see the number altogether was 112 here and 189 here, and only 8 of them overlapped, which means only 8 of the proteins that we could detect in the secretome of pancreatic cancer cells could also be found in the patient sera. And these are the ones being shown here. So of the 189 informative proteins ordered here according to I don't know what numbering system, you can see these 8 behaved reasonably well as markers individually. Though looking at the training set on the left and the test set, which shows you also why you need just a training set, but also a test set to actually evaluate the performance of any markers you may identify, you can see that these 8 markers behave individually no better than all the others we found. For instance, the ID1, which is one of the best ones in the training set, was one of the worst ones later on in the test set. Nevertheless, it was informative, but the more interesting part was if you put a signature together, of these 8 markers, the 8 markers that we found both in serum and the secretome of tumor cells, we get an ASUI value, which is kind of accuracy of 95%. So with 95% accuracy, we can tell this serum comes from a healthy person or a person who has chronic pancreatitis or a person who has pancreatic cancer. Any other serum marker, any other mixture of 8 serum markers put together, never mind, for all of the 189, any combination, this was the best in performance we could obtain, which is 84%. Which means these markers, which show up in the serum and the secretome, are much better than any marker that only shows up in the serum as it is. And as a matter of course, what we have also secretome data on the activated stellar cells, which are tumor-specific, tumor-associated fibroblasts, which are different to normal fibroblasts, that is, and 2 macrophages, which are actually infiltrating pancreatic tumor tissue a lot. And of course, also stem-like tumor cells of PDAC, of pancreatic cancer. And with these secretomes, added what we think, and this is again work ongoing to identify actually a signature of a few proteins, doesn't need to be 8, never mind, we'll see how many it will be, that actually performs better than the 95%, although 95% is pretty good. But you have to keep in mind, even 99% is not that good for, let's say, screening. Because 1 in 10,000 people will have pancreatic cancer once in his or her lifetime. But if you pick 10,000 people, basically none of them will have one, because the one who may have it 10 years from now or 15 years from now, and so on and so forth, which means at best there is 1 patient in every 10,000 people. But if you have a 99% accuracy, you would call 100 of those people and call them that they may have pancreatic cancer, which of course is not good enough. Calling 100 people, telling them, you may have a deadly disease, or while only 1 of them does actually have it, or probably even less than 1 on average, this is not the way to achieve what we want to achieve. For that matter, we apply this nevertheless, because what we can apply it for is actually to diagnose tumor recurrence, which means what we have is blood samples taken different periods prior to recurrence of the tumor. Once the tumor has been resected, as I said, 20% can undergo resection of the tumor, but usually they have recurrence within about 2 years, some a bit longer, but on most patients, unfortunately, have recurrence of the tumor in 2 years. And that means, we are able to pick it up, hopefully earlier than that and earlier than current markers. Currently, there's basically just CA99, which is 1 marker, which is pretty bad specificity and sensitivity in the range of 70%. Something like this. And what we find is actually we have significant variations in the amount of protein, of various proteins in the blood of those patients. And as it looks, again, this is not yet finished, not yet published, it looks as if we are not just being able to say, well, this patient does have a tumor again. So there is a recurrence. We can even predict when the recurrence is taking place, which means directly after surgery, a few weeks, a couple of weeks afterwards, a blood sample has been drawn. And from the data in this blood sample, we seem to be able to predict when the recurrence is going to happen in this particular patient with very good accuracy. Again, we are currently using a clinical trial, or having a clinical trial with our clinical partners to see how well this actually works in real life. Last but not least, we also look at cells, or actually not cells, but all kinds of cells and the proteins in the cell surface or at the cell surface. And the way we do it is the same antibody array that I've just presented to you. What is different in that respect that we're actually using blood and take the living cells from the blood and apply it to the array. And then what is happening is shown here that certain proteins in the outer membrane of the cells are actually binding to the respective antibody, which is on the array. And then what you get is shown here on the right, some binding of cells to a particular antibody. Well, as a matter of fact, what you see here is individual cells. So each point in this graphic here represents a single cell. That means we are working quantitative. We can count. So which means, for instance, at the left here, you see there are 4 cells binding. Here it is 5 to 6. And I don't know, can't remember, it was 12 to 15 in these. And more here, which means we can really count the number of cells binding to a particular antibody from each sample. So which means we are quantitative. And in terms of sensitivity and specificity, you can see here, for instance, monocytes, M1 macrophages and M2 macrophages to this particular antibody, only the M2 antibodies, macrophages do bind, which means we get a lot of information. For instance, looking here at some of the markers that we have, what you see is, none of the M1 macrophages is binding to any of these antibodies binding to this particular CT marker, while all M2 macrophages do bind, and with a reasonable distribution like this, which means we can very nicely discriminate between them. And shown just as an example in comparison to a flow sorting analysis here, this is a microarray data. You can see they are very equivalent, but of course we cannot just look at one cell type at a time, but more or less to all cell types in parallel, just applying the whole blood sample as it is in some diluted form, of course, to the array and see what is binding, subsequently labeling it with another fluorescence dye and seeing the signals, the kind of shown here. Never mind that. Going on to the last part, just to show you how proteomics may actually improve treatment options in future. And actually we came from a very different area into this, which is synthetic biology, or, synthetic biology, which means we talk nothing natural, but non-natural kind of molecules we are looking at. And what we are mostly interested in are, of course, DNA and protein. And what you see here on the left is a normal ddNA conformation and a protein, which is actually in the L conformation. This is how nature made it happen. But in principle, of course, what exists or could exist is the same protein in a mirror image form in the then D form. And the DNA could actually exist in a L form, which is again a mirror image of the natural kind of DNA. A double helix then wouldn't turn to the right, but would turn, and actually does turn to the left, because what we can do is the molecules on the right, we can synthesize chemically. Unfortunately, not yet enzymatically, but this is one of the issues we are going to tackle, and others as well, as a matter of fact. Once you think, what does it matter in terms of treatment? Well, for once, what we set out, as said, was really to understand the whole thing. We synthesized one enzyme, DNA ligase, which enables to link DNA to larger fragments, as shown here. We succeeded in doing this. We also worked on another couple of molecules. And one interesting aspect of this is that proteins made of D amino acids are not biodegradable, which means none of all the proteases being around is degrading the protein, the D protein. Let's say this ligase on the right, the D version, which is, as you can see here, a mirror image really of that one, is being degraded by proteases, which means if you have a protein truck and bring it into the body, it's not going to be degraded by proteases that are present in the body. So far, quite a few proteins have been synthesized, all of them, as said, chemical synthesis, because what's missing mostly is really, the ribosome. That's too a complex structure to be easily formed or created artificially by chemical synthesis, since you also need a lot of RNA and proteins for that matter. As said here, if you look at the number of proteins, it's not easy to synthesize all of them. And then, of course, the structure should assemble appropriately to have the activity. And then you would need the cofactors really to get the ribosome activity. People work on this. we also a little bit, but to succeed, it's not going to be easy. What is interesting though is that many drugs already in use, protein drugs that is, actually are antibodies or antibody-like structures shown here, for instance. What they do is, or most of them, show that they trigger an immune response in the patient, which may actually make the treatment unfeasible simply because it doesn't do any good, but only harms the patient. Nice thing again about the D proteins, they are not immunogenic, which means they are no subject to T cell response, which means there will be no strong immune reaction to a D protein. So in terms of what we want to do is that, let's say we want to have binders, antibodies that are used as drugs, but made of D amino acids. And what we set up to do is actually, that we want to create a molecule that behaves like an antibody. It's a DARPin, which is a different structure. The advantage is that it's small, so chemically more easily to be synthesized. We do this in collaboration with Phil Dawson at Scripps and Andreas Plückton in Zurich, at Zurich University in Switzerland. And the idea is to identify a binder, not from the L version, but from the D version that specifically binds to this target protein, shown on the left. Well, the way this needs to be done is a bit complex, but it's possible. So the way we do it is that we actually take an L-DARPin library, a highly complex library. So Andreas Plückton has libraries that have a complexity of 10 to the 17, 10 to the 18, which is basically identical in complexity to the human immune system. And then what we do is we all have started and have been doing, our ongoing doing, is actually synthesize chemically the mirror image form of this natural protein, the target, or part of it, doesn't need to be the entire one. Then use this to select from this recombinant L-DARPin library, antibody basically library, the one molecule or the few molecules that binds very specifically to the D protein. Then we synthesize chemically a mirror image form of this binder. And this binder by definition, which is still something we need to show, exclusively, is that this binder is then really specifically binding the L protein, which means after all this processing, what we would end up with is a target, a natural target protein, and we would have a binder, an antibody-like structure that binds specifically this target, but it consists only of D amino acids, which means it's not biodegradable. It will last in the body pretty long, or much longer than an L protein that is. And also it doesn't trigger any immune response, which means the patients will not, be resistant against the drug for the simple reason that the immune system will not be triggered to work at this drug. With this, wrapping up, we are pursuing two directions in this kind of work. For once, we want to identify functional proteins, like, for instance, also inhibitors that we could synthesize chemically and, in a mirror image form, but nevertheless binding the natural molecule, of course, and thereby having, let's say, very stable inhibitors, like, for instance, the DRD2 form, that would be something to be interested. And also, we and others really are aiming at towards an enantiomeric, self-replicating system, as mentioned before. Really, the ribozyme would be the most important part of the activity of the ribozyme, not the molecule itself, but an activity that allows translation of RNA into protein. And performing protein synthesis is definitely needed, because then we would have a self-replicating system and could create even large D amino acid proteins very easily. This is the future. I'm sure it's going to be part of the future at least. And with this, I'm done, but not without, thanking the people who do the work that I then talk about usually. So Andrea Bauer is the person who is having most of the connections with clinical partners of ours, therefore is responsible for obtaining the samples, characterizing them, and so on and so forth, is organizing all this kind of work. Mohammed is the person behind the antibody array that I mentioned. Yenan is the person with the methylation data analysis, ribosome binding, something I will talk about tomorrow, as mentioned. Then Henning is the person behind the protein arrays, where we look at 3,000 human proteins and, let's say, antibody binding from sera to these proteins. And last but not least, Favas, who is the guy actually performing the recurrence analysis, which means predicting the occurrence of tumor recurrence in pancreatic cancer. Currently busy with performing this clinical trial analysis, and as I said, hopefully by the end of this year, spring next year, we should be able to tell whether what we have in data actually is true in real patient applications. And of course, that would make a difference at least to those 20% of pancreatic cancer patients that undergo surgery. With this, I'm done. Thank you very much for your attention. And I will be happy to answer any questions you may have. Thanks a lot. With this, as said, I'm done. And as said, happy to answer questions that you may have. Thank you.
Thank you very much, Dr. York. That was a very, very impressive talk, and we learned a lot. And I hope that there will be a lot of questions regarding this these very nice studies that you are conducting. And I would just like to remind here that after the 2 more talks to go, we'll take all the questions from the audience. So with this, I would like to introduce you all to our next speaker, Dr. Marcela Hill. He is the Associate Professor at the Institute Pasteur de Montevideo, Uruguay, and he's the Director of Center for Translational Immunology. And his main interests in research are around immunoregulation in cancer cells and dendritic cells, and particularly with the focus of immunotherapy. He has many patents to his credit, and we very much look forward to listen to his talk today. And the title of the talk is TMEM 176A/B, ion channels as targets in oncology. Over to Dr. Marcelo.
Welcome to this presentation. My name is Marcela Hill. I work at the Institute Pasteur in Uruguay in South America and at the Faculty of Medicine at the University of the Republic. I'm going to thank the organizer for their kind invitation to this meeting, and I'm going to talk about these emergent ion channels as potential targets in oncology. And I have to do a disclosure with Ardan Pharma, which is a startup which we have created from technology with TMEM176A and B inhibitors. So TMEM176A and B are members of the transmembrane family MS4A, from which the most known member is CD20. TMEM176A and B are ubiquitously expressed in normal and in cancer tissues, and within the immune systems, they are highly expressed in CD11B positive conventional dendritic cells, macrophages, innate lymphocyte cells, and ROR gamma T positive cells. At an intracellular level, they are localized at the phagosomes and the Golgi, and we and others have associated its expression to immune tolerance. We have described that these molecules are non-specific monovalent cation channels, and we have recently shown that these ion channels can inhibit inflammasome activation through ionic mechanisms. So the MS4A family is composed by several members. As I said before, the most known is CD20 or MS4A1, and these molecules are either ion channels or modulators of immune receptors. And as you know, CD20 is a calcium ion channel, and these molecules, TMEM176A and B, are monovalent cation channels which acidify the phagosomal lumen and the Golgi network. But which is the role of these molecules in cancer? So if we analyze the cancer cycle, you know, this process by which tumor cells die and then dendritic cells pick up antigens, present them to T cells, and this adaptive immune response will kill the malignant cells, starting again this cycle. TMEM176B and A play an important role on one part in dendritic cell biology by modulating antigen processing and inflammasome activation, but today I'm going to focus on the role of TMEM176B in the malignant cell. So in both parts of the cycle, in regulating dendritic cell biology and malignant cell, TMEM176B can be target for pharmacological agents to modulate this cycle. When we started to work with these molecules, we started to work with dendritic cells, and we were focused on antigen processing mechanisms. As you know, CD4 positive cells, T cells, recognize antigens when they are presented in class 2 MHC molecules. And CD8 T cells can recognize tumoral antigens which are presented by the cross-presentation pathway. We got interested in these pathways because TMEM176B and A are highly expressed in these structures in endosomes and phagosomes, which are dealing with these antigen processing mechanisms. So in this paper, we analyzed wild type and TMEM176B knockout dendritic cells, and we analyzed division proliferation of CD8 T cells specific for OVA antigen. And we observed that if we use dendritic cells from the TMEM176B knockouts, then we had diminished activation of the antigen-specific T cells as compared to the wild-type dendritic cells. However, if we treated these dendritic cells with the already processed antigens with a peptide, which is recognized by these T cells, we observed no difference in stimulation of the T cells when we compared wild-type and TMEM176B knockout dendritic cells, strongly suggesting that this molecule was playing a role in antigen processing when stimulating CD8 T cells. Phagosomal pH is a known important process in this antigen processing mechanism. So we analyzed phagosomal pH in wild type and knockout dendritic cells, and we observed that the TMEM176B knockout had a strikingly alkalinized phagosomal pH, suggesting that this molecule was regulating phagosomal pH in dendritic cells. Since this process of regulation of phagosomal pH is tightly regulated by ion channels, we speculated that this molecule could be indeed ion channels, and we worked with Xenopus oocytes, and we overexpressed TMEM176B, and we observed an inward current that was measured when we overexpress this molecule and acidify the medium of these oocytes. And this was almost completely blocked when we substitute the sodium ions with the non-permeant NMDG. So these TMEM176B and A are transporting sodium in the phagosomes, and in that paper we also showed that this ion transport is important for acidification of the phagosomal lumen by the vacuolar ATPase, and these mechanisms are explaining this altered phagosomal pH in the knockout dendritic cells. But there is a process in myeloid cells, in dendritic cells, macrophages, which is tightly regulated by ions, and it is inflammasome activation. So inflammasomes are cytosolic complexes that regulate the activation of inflammatory caspases, such as caspase 11 and caspase 1, and they can cause pyroptosis, which is an inflammatory form of cell death. And they can also, through activation of caspase 1, they can also regulate IL-1 beta and IL-18, triggering, producing the active forms of these inflammatory cytokines. And caspase 1 and caspase 11 are also cleaving Gas-Dermin D, leading to pores which in the membrane, which can lead to this inflammogenic form of cell death, which is pyroptosis. So we speculated that TMEM176B and A as ion channels could be regulating this process because inflammasome activation and caspase 1 activation is known to be tightly regulated by ion fluxes, mainly the cytosolic content of potassium and calcium. So we generated this TIM176B knockout mice, and we injected these mice with ATP in the peritoneum, and we studied the recruitment of neutrophils in these animals. And this model is known to depend on inflammasomes and on caspase-1 activation. And we observed that in the TIM176B knockout, we had a huge recruitment of neutrophils, and this was almost completely inhibited in the double knockouts with caspase-1, showing that this neutrophil recruitment was due to enhanced caspase-1 activation in the TIM176B knockout mice. I'm not going to get into the details in this, but we showed that this enhanced inflammasome activation was due to ionic mechanisms. So in these knockout mice, we had increased calcium, and this was we show in this paper that we had increased calcium and increased potassium efflux in these knockout dendritic cells. But which is the role of TIM176B in cancer? So early this paper from Koshunko and colleagues showed that TIM176B and A and B are highly expressed in several normal and tumors, but the role of these proteins in cancer biology was not known at this moment. So trying to understand how TIM176B could control tumor biology, we tried to correlate the TIM176B expression in colon cancer to overall survival. So we performed staining with anti-TIM176B antibodies, and we observed that we have staining in the parenchyma and in the stroma, and we identified within several colon cancer tissues that there were some low expressors and high expressions in the parenchyma and also in the stroma. So we tried to correlate this expression to overall survival. When we analyzed parenchymal expression, we observed that there was no correlation with overall survival with high and low expressors. However, when we analyzed the stroma, we observed that high TIM176B expression was associated with poor overall survival in these colon cancer patients. So suggesting that TIM176B could be modulating antitumoral immune responses in colon cancer, and we have directly shown this in this paper. I mean, a role for TIM176B in controlling anti-tumoral immune responses. But as I said before, today I'm going to focus mainly on a potential role of TIM176B in malignant cells. So in this paper, these authors found a correlation between high TIM176B expression in gastric cancer and poor overall survival. So first, they observed a high expression of TIM176A and B in gastric cancer as compared to normal gastric tissue, and high expressors again were associated with a poor overall survival, suggesting again that TIM176A and B could be playing a protumoral role, in this case in gastric cancer. We then started to work in chronic lymphocytic leukemia, and we correlated TIM176A and B expression with overall survival, and we observed that there was a correlation between high expression of TIM176B and A with poor overall survival. So this was at the RNA level, and we also analyzed protein expression in indolent and progression patients, and we observed that for TIM176B, progressor patients showed a high expression of TIM176B versus indolent patients. And you can see here the Western blot, so this was true for TIM176A, but we found no correlation with TIM176B. So TMEM176A is significantly overexpressed in chronic lymphocytic leukemia cells from progressor patients. So taking into account this regulation of inflammasome and caspase-1 activation that we have previously shown in myeloid cells, we speculated that also in chronic lymphocytic leukemia cells, TMEM176A could play a role as an inflammasome inhibitor, so controlling activation of caspase-1 in chronic lymphocytic leukemia cells. So we observed, we analyzed caspase-1 activation in indolent patients and in progressive patients, and we observed that in progressor patients there was an inhibition in caspase-1 activation, and we found a significant difference between progressive and indolent patients when studying active caspase-1. So cells from progressor CLL patients have lower caspase-1 activations when comparing to indolent patients. So we think that in these patients, TMEM176B is inhibiting inflammasome activation. So we wanted to directly show this hypothesis. So we performed a knockdown of TMEM176A with a small interfering RNA, and we studied caspase-1 activation. And we observed that using two different eRNA to knock down TMEM176A, there was a significant increase in caspase-1 activation, showing that also in chronic lymphocytic leukemia cells, TMEM176B is inhibiting inflammasome and caspase-1 activation. So we speculated that TMEM176A could be inhibiting pyroptotic cell death in chronic lymphocytic leukemia. So to study this issue, we went to study a fragment of Gasdermin D, which is associated with pyroptosis, because this fragment forms the pore in the membrane, in the cellular membrane, leading to the pyroptotic cell death. So we performed Western blot studies assessing Gasdermin D P30 fragment, and we observed a nice correlation in the sense that in patients where we observed high TMEM176A expression, there was low cleavage of the pyroptotic fragment of Gasdermin D, and the opposite was true in low expressors of Gasdermin low expressors of TMEM176A, we had big expression of the pyroptotic fragment of Gasdermin D. And here you can see the inverse correlation between these two players, TMEM176A and the pyroptotic fragment of Gasdermin D. So what we think that is going on here is that in progressive or aggressive chronic lymphocytic leukemia, we have high expression of TMEM176A, which is inhibiting inflammasome activation and controlling pyroptotic cell death of the tumor clone. So we developed this compound AP1, which is a strong inhibitor of TMEM176A and B. So we treated this chronic lymphocytic leukemia with this compound, and we observed that we were triggering cell death with active caspase-1, and this cell death was inhibited when we used a caspase-1 inhibitor. So AP1, the TMEM176A inhibitor, increases cell death with DAPI positive and caspase-1 positive primary chronic lymphocytic leukemia cells, and this difference was lost when a caspase-1 inhibitor was added. And a similar result was observed when we analyzed the pyroptotic fragment of Gasdermin D P30. When we treat the cells with AP1, we trigger the cleavage of Gasdermin D. So inhibition of TMEM176A by AP1 produces death of leukemic cells through pyroptosis. We also have in vivo data, so we worked with this aggressive CLL model in mice, TCL1. So here we treated the mice with our compound, with the inhibitor of TMEM176A and B. We observed a significant survival in the treated mice compared to the untreated animals, showing that in vivo these mechanisms are also relevant, and this correlate with leukocyte levels in the untreated and in the treated animals. But other authors have also showed that TMEM176A can play a relevant role in other cancers such as glioblastoma. So in this paper, there was a high expression of TMEM176A and B in glioblastoma as compared to normal tissue at the messenger and protein level. And most importantly, there was a significant association of high TMEM176A expression with poor survival, strongly suggesting that these molecules may also play a significant role in glioblastoma biology. So in vitro experiments working with glioblastoma cell lines, these authors show that a knockdown of TMEM176A with two different scRNA triggered apoptosis of these cell lines, and this was associated with increased activation of caspase-3 and diminished expression of the anti-apoptotic protein BCL2. And at the signaling level, these effects of TMEM176A seems to be related to phosphorylation of RK1 and 2. They have assessed the phosphorylation of different kinases, as you can see here, but when they knock down TMEM176A, they observe a diminished phosphorylation of RK1 and 2, showing that TMEM176B and A could be promoting phosphorylation of these kinases, which are known to play a relevant role in inducing cancer cell proliferation and in inhibiting apoptosis. In vivo, in nude mice, we also showed that knockdown of TMEM176A was associated with diminished tumor growth in vivo, and here you can see the size of the tumors when TMEM176A was knocked down. So in vivo, these ion channels seem to play also a relevant role in glioblastoma. Nevertheless, there are some challenging results in other kinds of cancer, such as in hepatocellular carcinoma, because these authors have shown that TMEM176A may play a tumor suppressor role. So they observed that methylation of TMEM176A was associated to poor survival. And you know, methylation is leading to diminished expression of TMEM176A. So here they observe expression in adjacent tissue, but not in cancer cells of the TMEM176A, and this was a significant diminished expression in cancer versus adjacent tissues. And there is a negative correlation between the expression levels of TMEM176A and the methylation of the promoter region of this gene. And we also showed in vitro using cancer cell lines that TMEM176A triggers apoptosis in hepatocellular carcinoma. So here they are overexpressing TMEM176A, and the number of cells significantly diminished as compared to control vector in two cell lines. and the opposite is observed when they knock down the protein with a small interfering scRNA, and in that case they increase the number of cancer cells when knocking down TMEN176A. And when looking at apoptotic cells, overexpression of TMEN176A triggers apoptosis, whereas the knockdown of this ion channel controls apoptosis in two hepatocellular carcinoma cell lines. So these results suggest that at least in hepatocellular carcinoma, and this group has also shown it in esophageal cancer and in colon cancer, that TMEN176A could play a tumor suppressor gene. But what about the mechanisms? So in this scenario, the authors observed that when overexpressing TMEN176A, they had diminished phosphorylation of RK1 and 2, and the opposite is seen when knocking down this ion channel in these hepatocellular carcinoma cells. And when overexpressing TMEN176A, there is an overexpression of a RK1 inhibitor, which is SER1A, and so the author suggests that at least in these cells, TMEN176A is leading to inhibition of phosphorylation of RK12. So as a summary, what we have shown is that TMEN176A and B can play a protumoral role, and this could be due to inflammasome inhibition and pyroptosis inhibition. So when using inhibitory compounds, we can trigger, as we have seen in chronic lymphocytic leukemia, you can trigger inflammasome and pyroptotic cell death. And another mechanism is to promote phosphorylation of RK12, and this kinase will lead to increased survival and increased cancer cell proliferation. However, it has also been shown that TMEN176A and B can play a tumor suppression gene role, but in that case, through mostly unknown mechanisms, TMEN176A-RB seem to be inhibiting phosphorylation of RK1 and 2. So we are working with these compounds, AP1, which is a TMEN A and B inhibitor, and we are carrying out physicochemical characterization, pharmacokinetic, and so far our results are good, and we'll continue characterizing these compounds as potential new tools to control cancer. We are also dealing with toxicity studies. We have done cardiac toxicity because these molecules were suspected to play a role in controlling cardiac electric mechanisms and morphological issues. But so far, we observe no toxicology problems with these compounds, and we continue characterizing the TMEN176A and B inhibitors. So our main idea is to continue the validation of the target, to continue working on the preclinical support, and to in 2023 to start a clinical trial with these compounds, and our main indications will be melanoma and chronic lymphocytic leukemia. So at the end, I would like to thank people from my team, Mercedes Segovia and Sofia Russo, Matthias Heldres, who worked on the characterization of Torid of TMEN176B as inflammasome inhibitor, and in the chronic lymphocytic leukemia studies, we collaborated with a team of Pablo Peso, also from Pasteur Institute in Montevideo. So thank you very much for your attention.
Thank you, Dr. Marcelo. That was a very good talk, and I really learned a lot from your work, and I see there's a huge promise in this kind of work that you are performing. So again, I would like to remind our audience that please type in your questions in the chat window, and we'll take all the questions at the end of the last talk, which I am going to introduce you. The last speaker of this session is Dr. Rob Ewing. He is the Director of the Graduate School of Biological Sciences, Proteomics and System Biology at the University of Southampton, UK. And his work focuses on protein interactions networks in cancer cells, which underlie critically important signal transduction pathways in many human cancers. And his interests are around the pluripotency of embryonic stem cells as well. So we very much look forward to his talk, which is on proteomic analysis of protein networks in colorectal cancer. Over to Dr. Rob, please.
Hi everyone, my name is Rob Ewing and I'm from the University of Southampton in the UK. It's a great pleasure to be here speaking with you at the symposium, and thank you again for the invitation. What I'm going to tell you about today is some of our work in using proteomics to analyze protein networks in cancer, and I'll mainly focus on the techniques to show you, how we do these experiments, but also give you some of the applications and the results that we've generated over the years. Here's a little bit about myself. So essentially, I've been interested in networks, molecular networks, for a good part of my career, and we've really applied both computational or bioinformatics techniques to analyzing those networks alongside techniques such as proteomics. And the core research question that we're interested in is, how do protein networks respond or change in response to human disease? So how are the networks altered, for example, in different disease states, such as in cancer? So in my research group, we really do several things, and these are the main themes. On the one hand, we contribute to the development of new proteomic and computational tools. So for example, I'll talk later about some of the work we've done in developing proteomics techniques for interactions. We're also very interested in how we analyze protein interaction networks, and for example how we can integrate different kinds of omics data, not just proteomics data. And we apply these techniques to several specific areas, and what I'm going to tell you about in this talk is our interest in colorectal cancer, and in particular the Wnt signaling pathway and its role in colorectal cancer. So we've been interested in defining new protein components of the Wnt signaling pathway, and I'll show you how we do that with our proteomic tools. So I'm going to start by really telling you, or giving you an overview of the techniques that we use for proteomic identification of protein-protein interactions, and then I will tell you a little bit about the biology and also some of the applications that we've made to discovering new components of the Wnt signaling pathway. So this is a very high-level overview of how proteomics is broadly used in cancer research. And what I've done here along this axis is I just laid out the kind of models or samples that we use in proteomics, all the way from cultured cells, for example, two-dimensional cultures or three-dimensional cultures of cells, for example, through model organisms to human samples of tumors or biofluids. And on this axis, I've put the kinds of experiments that we can do in proteomics. So we might, for example, be analyzing individual proteins, or we could be analyzing protein-protein networks, or post-translational modifications, or whole protein profiles. And the reason I'm sort of displaying this way is to illustrate the fact that according to what model and what technique we're using, we're going to get different kinds of data with different sort of complexity. So for example, if we take whole tumors and we use protein profiling, we actually get pretty complex proteomic data sets, in part due to, for example, the heterogeneity of tumors from patients. On the other hand, and this is mainly what I'll tell you about today, if we have simpler models, for example, immortalized cells or organoid models or 3D cultures, and we're looking, say, at protein-protein interactions, we're looking at a much simpler system. Okay, so typically this is the sort of area that most of our experiments are done in, where we've got fairly simple models of the genotype or the type of cancer we're interested in, and we're using proteomics to uncover protein-protein interactions and protein networks. So this is mainly what we'll focus on in today's talk. Now, just to sort of review what you might see in the literature around protein networks in cancer, I broadly sort of categorize sort of thinking in this area and studies in this area to either sort of large-scale studies where perhaps researchers have integrated different kinds of data. In this particular study, it's quite a nice study. They look across different cancers and identify differences between the molecular networks in those cancer types. So these are kind of large-scale studies, or more focused studies. And I've just put a couple of examples for our own research here, where we use proteomics to uncover new interactions between oncogenic proteins. So this is work we've done using the techniques I'll tell you about today. But the really important point here is that, in cancer, we know that oncoproteins are extremely important, and we know that in cancer, oncoproteins undergo many changes. So, for example, they might have increased expression. They may lose or gain new interactions, which changes their behavior in the cell. They may undergo altered subcellular localization. So we know that many oncogenic proteins or oncoproteins are shuttled to the nucleus. Beta-catenin is a good example of that, and that changes the cellular phenotype. And we also know that many of them have altered post-translational modification. So it's really important, and this is why proteomics is so crucial in this area, because proteomics can really tell us about all of these aspects of protein behavior. And so proteomics really has a role to play when we're trying to tease out protein networks in cancer. So another perspective on this is really to think about how proteomics sort of interfaces with genomics. So over the years, obviously, as genomics technology has picked up in pace and accessibility, we've seen lots of studies that produce data like this. So this is taken from a study some years ago now, but it's a breast cancer study, and essentially this is genome sequencing and profiling of the mutations. These are the genes here, and these are the samples here. This is profiling of all of these somatic mutations across different patients, and these are very, very informative studies. But I think one role that that proteomics can play in these kind of studies is actually to start looking at what these somatic mutations actually might be doing in the cell. And so when we think about exploring protein networks and looking at protein interactions in cancer, we often think of this kind of data and asking this question: what happens in the cell when the cell has, for example, a given somatic mutation? That's a question we can ask by studying the protein networks. This slide, what I wanted to do is just explain sort of the broad different approaches that we have in proteomics for analyzing protein networks, and I like to think about those approaches in two ways. The first is we've got techniques that I'll mainly focus on today that are involved in studying the physical interactome. So that's really, which protein is interacting with which, and we might start with a target or a bait protein and then be able to identify the direct protein-protein interaction partners. Or we might use other techniques, such as protein profiling, to kind of uncover what we can call the functional interactome. So these might be proteins that aren't directly connected, for example, in the network, but which have some kind of functional connection. So experiments that might enable us to do this might be, for example, through induced mutation or targeted knockout, that kind of experiment. But mainly what we're going to talk about today are experiments like this, where we want to actually uncover the physical interactions for a given target protein. So let's turn now to thinking about the proteomic techniques themselves. What I'm going to do is describe the technique that we've used most, which is the technique for interaction proteomics, for detecting protein-protein interactions, called affinity purification mass spectrometry. And the essence of the technique really is that you have a target protein called a bait, and that bait protein you can detect using an antibody or an epitope tag. But in essence, you use the antibody against the protein or its tag to semi-purify a protein complex. So this is the step of affinity purification. So by purifying a protein complex from the cell of interest, you can then subject that protein complex to mass spectrometry, and from the mass spectrometry you can infer which of what the prey or the partner proteins are for your target of interest, and this allows you to build up protein networks. Now, one sort of drawback of this technique that I'll tell you a little bit more about in a moment is that how you distinguish true positive from false positive interaction partners. And in essence, what you get in these techniques, you get non-specific binders, for example, in the affinity purification that actually end up in your mass spectrometry and may give you false positive results in your protein network. So we applied this technique on a large scale actually some years ago in a biotech company, and our intention was to map the protein-protein interactions for over 500 human bait proteins. We took those 500 bait proteins bait proteins and we expressed them in human cells, and then did the immunoprecipitation using an antibody, and then went through the steps of mass spectrometry to identify all of the partner proteins for each of our starting preys, shown here in blue, starting baits, shown here in blue. And we identified approximately 8,000 prey proteins and 10,000 protein-protein interactions. So these techniques are very powerful. They can generate a lot of data, but there are some drawbacks that I want to just talk a little bit more about in a moment. So one of the limitations, as I said earlier, is that you can pick up on contaminants. And if you don't filter out these contaminants, then you end up with a protein network that might have false positives. And so one of the pieces of work that we did a few years ago was to generate a repository. This is called the crapome, and essentially it's a repository that contains all of the contaminant data from these kinds of experiments. So what this means is that a researcher can take their list of protein-protein interactions and they can essentially upload them into the system and identify which ones are most likely to be false positives, so that they get a better list of true positive protein-protein interactions. Now I want to move, now I've explained a little bit about the kind of techniques that we use. I want to just go into the last section and tell you about the applications that we've made using these techniques in colorectal cancer and in the Wnt signaling pathway. So for several years we've been interested in defining new components of the Wnt signaling, and particular components that are relevant to colorectal cancer. So we know from the work of Hans Clavers and many others who've contributed to this that Wnt signaling is very important in the normal homeostasis of epithelial cells in the gut lining. So this is a cross-section of the mammalian gut, and we know that the process begins down here with stem cells. These stem cells differentiate and migrate up the epithelia, the walls of the villi, and eventually they're sloughed off after about 3 or 4 days. And this process is like a conveyor belt in the healthy gut, but in the diseased or the gut that's going to develop colorectal polyps or tumors, what can happen is that you get formation of a tumor, and these are often caused by a failure to inactivate Wnt signaling. So our interest over the years has really been to try and target some of the known players in Wnt signaling and identify novel protein-protein interactions. And I'll tell you about some of the work we've done in this area. So just before I go onto that, I just want to explain a little bit more about how we do these experiments and the role for us that genome engineering is now taking in our studies. So what I told you before about interaction proteomics was taking a bait protein and identifying the interaction partners for that bait protein. What we do more frequently now is to actually do some kind of engineering in the cells, because of course genome engineering really allows us to produce tags within the cells that we're interested in. So for example, we could take a cancer cell and you could induce a mutation in a given locus, or we could add a tag onto a target protein, as illustrated here, an epitope tag, and then we could do our proteomic analysis. And this is more of the workflow really that we've been most interested in. These were my collaborators a few years ago when we were developing these protocols. But essentially this kind of approach, we call it knock-in APMS, allows you to have physiological expression of the bait proteins. Obviously, if there's too much of the bait protein, you might get false positive interaction partners. It's also applicable to many cell types, and as I'll show you, we can apply this to cancer cells. So what we've done over the years is to build up a collection of engineered cell lines, and there's just some examples here drawn from the Wnt signaling pathway. So for example, we can do selective knockout. You could have a cell in which you've got a wild type and a mutant cell and knock one of the alleles out, and then compare the mutant and the wild type cancer cells to look at interaction partners. We've also got projects where we put tags on individual cells in a given genotype. So this really allows you now to recreate kind of genotypic models that you might encounter in a given tumor, and then to investigate those in detail. And just in the last couple of minutes, I just want to share with you a couple of projects where we've used these techniques. So this is some of the early work we did in this area. What we were doing here was looking at the adenomatous polyposis coli, or APC tumor suppressor. This is a very large protein, which means that there are variable quality antibodies. It's difficult to work with. So we used the knock-in approach to take colorectal cancer cells, to put a tag on the APC locus, and then to use that interaction proteomics protocol that I described a few moments ago. And to cut a long story short, what we can do when we do these experiments is pick up on a lot of the known factors that already are known to interact with APC. So for example, the Wnt signaling factors here. And we're able to show that even just in a few experiments, our particular approach of engineering the cells first and doing these experiments actually was really in a very effective way of picking up on protein interaction partners. So we now have a good collection of APC protein-protein interactions, and of course that really helps us build up a picture of how the destruction complex in Wnt signaling is actually working. This is another example. We've got a couple of different projects in this area. It's not strictly Wnt related, but we were interested in the DNA methyltransferase, and we took colorectal cancer cells and knocked in a tag onto the DNA methyltransferase. So for those of you who may not be familiar with this, the DNMT1 or DNA methyltransferase is a very important protein for regulating methylation across the genome and is often dysregulated in many cancers. And we discovered actually how DNMT1, in doing these experiments, how DNMT1 is actually regulated through a deubiquitinase called USB7. We've actually done some subsequent work with DNMT1 to show that it interacts with the beta-catenin interaction, the beta-catenin protein from Wnt signaling as well. So these techniques are really broadly applicable to discovering new interactions or indeed different ways that these proteins are actually regulated in cancer. So this is just really the sort of final slide, just to squeeze a few more results in. But this on this slide, what I've done is put some of the data from a novel interaction that we discovered a few years ago between the DNA methyltransferase and beta-catenin, which is the central effector of the Wnt signaling pathway. And I won't go through all the data here, but I think the point of this slide is that very often, when you discover these protein interactions using proteomics, you also need to validate them using more targeted approaches. And so, for example, we've done various experiments here, such as doing knockdowns using siRNA to confirm actually that there is an interaction, or doing immunocolocalization in the cells. These are in colorectal cancer cells, showing that actually there is co-localization of the DNA methyltransferase and beta-catenin in the nuclei. And it turns out that the interaction between these two proteins is a cancer-specific nuclear interaction. These techniques do have the power to identify novel interactions. So I'd just like to finish there. That's a bit of an overview. I've told you something about the technology and also some of the findings we can get using those techniques. I'd like just to highlight several collaborators and colleagues. Jing Song, who used to be a postdoc in my lab, John Wang, who's a collaborator, Paul Skip, who's our proteomics director here in Southampton, Emily Bowler, who was a former PhD student as well. And I'd like to also acknowledge my current affiliation, University of Southampton, and previously when I was in Case Western Reserve University School of Medicine in Ohio, and also thank all of the funding bodies that have contributed to this work. Thank you very much for listening, and I'll be glad to take questions after the session.
Thank you very much, Dr. Rob. It was very nice presentation, and a lot of potential in all the work that you are doing. And we hope to see really good studies in the future based on these study ideas and lines. So with this talk, we get to the end of the session. All the talks are through. So we'll take the questions right now. And I would like to welcome all our speakers who I can see now. Thank you very much for joining us, and thank you very much for your for sharing your knowledge with us, which you have gathered through the years, and we learned a lot today from those. And I would just read few of the questions to discuss here. So one of the questions is from Dr. Rodney, and this is regarding, it's a something regarding the gap between the basic and clinical research, and the audience is asking that these are the two ends of a continuum. However, in Pakistan and lower middle income countries, so there is a divide in these specialties, and we struggle to establish translational research units which can take research from bench to bedside effectively. And basically, this is connected to this a point that you raised in your talk, that comprehensive screening can be harmful. So yes, we offer the clinical, testing, screening, BRCA1, BRCA2 testing. You have the result. And how ready is the patient to go for the mastectomy based on that information? How much counseling is needed for the patient? So all that work which is committed to find the results, that these genes are important for screening. What are your thoughts on the importance of counseling given to the patient?
Can you hear me okay?
Yes, we can.
Oh, great. Well, for any woman who wants to take up the notion of screening, she does have to have counseling about the implications of the screening test that she's going to have. The issues are what sort of test is going to be offered and what are the implications of that test. If we look at what we know with a little where we have more information, and that is with primarily BRCA1 and BRCA2 mutation carriers, and we've been studying those for the last 30 or so years, we have gained a lot of information as to what it means to be a carrier of mutations of those genes. The counseling, therefore, is centered around what we know in terms of that woman's particular disease risk, what sort of family history she has, and what is the probability of her presenting with breast cancer, ovarian cancer, or another malignancy. Where it all comes apart is with these new genes that we've identified, where we really don't know what other malignancies are associated with them, apart from the fact that it's breast cancer. So the risk for a particular disorder is quite obvious, but when we begin to stretch it out to what we've observed, especially in BRCA2, it becomes a little less consistent. So you've got to be very careful with the counseling. You don't want to give the person undue expectations. You want to give her a realistic appraisal of what risks she has on current information. If there is a change to that information, ideally you'd like to reappraise that woman of the risks that are now available or understood about disease in that context. So the idea of consultation and risk assessment for any woman, really, if in an ideal world, and unfortunately we don't live in one, is a continuous assessment. So as things change, she should be called up and offered the opportunity to hear about new findings in the field and the ability to adjust her care over time.
Yeah.
As we sort of learn more about these genes. Okay. Does that answer the question?
Yeah. That's absolutely, yes, that's very good answer to what was asked, and I hope that it helps them. So the next question is also based on your study, Professor Scott. He says, if some founder mutations are detected in a population, it would be better to test those mutations first before gene panel testing, or go straight for gene panel testing? Thanks.
That's really a function of how common those founder mutations are. For example, in Poland, where there are very high frequencies of founder mutations, it makes sense to screen those because a significant proportion of women have changes in those genes. If, however, it's a relatively low percentage, but it is a little bit more frequent than you'd otherwise expect, then there's really no advantage to doing it, because if you don't find anything, you're then going to have to screen anyway. So you may as well do it altogether. The advantage of next-gen sequencing and screening women for causative changes in a variety of breast cancer susceptibility genes is the more you do, the cheaper it becomes per individual. So to start pulling them apart and saying, well, we'll go for a founder mutation, and if you don't have it, we'll come back and do a gene panel test, sort of defeats the object of the exercise, because the more you do, the cheaper it becomes. So my recommendation would be, if it's not a high frequency in a given population, do a gene panel test straight up and don't worry about the founder mutation within a given population. If it's, say, less than 10 or 15%.
Okay, well, thank you. Thank you very much. My next question is from Dr. York. Very, very interesting talk. And this is this is my question actually, that you did the work with all those 8 markers in the secretome and in the serum itself. Did you try to see the expression of those 8 markers in the tissue itself? You know, because the tissue from, you know, where the disease is actually happening. You know, for example, if you take the tissue slide on from a paraffin embedded block, did you see the expression of those markers in the parent tissue?
Well, we have that data, but we didn't look at it in particular, since, you know, with all the data that we have, there's no apparent correlation between what you see in the secretome or in the serum of the respective patient, or the concentration of the proteins in the cell. So the intracellular concentration of proteins is in most cases very different to what you see from the secretome of these cells, never mind, or of the tissue that is, or of what is being seen in the serum, which means there's no correlation whatsoever. And therefore, you know, the markers that may work in serum are likely not to work in tissue. And also, once you have tissue at hand, it's easy to diagnose a tumor and also to do a prognosis. So we wouldn't gain anything from this knowledge, but we do have the data, all right, yes.
Okay, okay, thank you. Thank you very much. My next question is from Dr. Marcelo, one of the audience. They typed in, and I'm just going to read this. So it says that, can a targeted approach to silence TMEM expression in tumor microenvironment be developed to manage its effects?
So, can you hear me?
Yes, we can.
So it's a very good question, because actually we are working on encapsulating MEM176B and A inhibitors in nanoparticles. So through this approach, we can couple antibodies to these nanoparticles and direct these inhibitors either to the immune system or to malignant cells. So in the cases where these proteins work as tumor suppressor genes, it will be actually dangerous to inhibit it in malignant cells. But we have to keep in mind that these proteins play a role in the malignant cells regulating either survival and proliferation, but they also play a role in leukocytes. And most of our work has been focused on this side of the equation, on looking at how these proteins can impair anti-tumoral immune responses. So through this approach, I mean, by encapsulating these inhibitors in the nanoparticles, the idea is to direct the inhibitors to the leukocytes. And here again, we can specifically target these nanoparticles to specific subsets of myeloid cells. But through this approach, we can modulate anti-tumoral immune responses. And the idea is to keep safe, you know, the malignant cells, do not inhibit these proteins in malignant cells, at least in the tumor, where these proteins seem to play an anti-tumoral role. So indeed, we can do it, and we are working on this possibility and in coupling specific antibodies to these nanoparticles.
Okay, okay. Well, thank you very much for your answer. My next question is from Dr. Weisel. regarding the proteomic analysis, can a method to obtain samples from pre-tumor microenvironment be developed? Do you think this would be a better option for early diagnosis screening?
The answer is yes and yes. I mean, there are methods to obtain tissue samples using biopsy, but obviously this is pretty invasive, so it should be avoided as much as possible. Once you have tissue, and it has to be the right tissue, you should hit the tumor, which you sometimes don't. But anyway, I mean, that's pretty up and running, so that's working. And once you have tissue, the diagnosis, as mentioned before, is usually quite straightforwardly, also at the molecular level. So there is a method in place. I don't know about a better method yet, but of course it's an invasive method, which means you should try to avoid it. And that's the whole reason why we do this liquid biopsy kind of approach.
Liquid biopsy, yeah, absolutely, yeah. Thank you. The next question is from Dr. Rob Ewing. So it's regarding the dopamine receptor work. Is there any association between mutations associated with schizophrenia and reduced enhanced association for pancreatic cancer, or are the numbers too small to draw any conclusions?
Well, the quick answer is we don't know. We haven't looked at this at all. So I have no idea.
Okay. So I hope it answers everyone's questions. Since there are no more questions here, so I thank you all the speakers to joining us and giving us very wonderful talks, and we look forward to the second session of cancer research, which is going to start in less than 2 hours. So hope to see you again, and thank you all the audience for joining us today. It was a very good session, and with this I say goodbye to you all. Thank you very much.
Right. Bye-bye.
Bye.
Bye.
Bye. Bye. Bye. Good morning and good evening and hello to everyone. Thank you for joining us at the 20th Shaukat Khanum Cancer Symposium. And this is our second session for the cancer research. And I am Saira Salim, and I work as a research scientist at the Basic Sciences Research Department at Shaukat Khanum Cancer Hospital, Lahore. And I am very thankful to my speakers and audience to join us for this session. And I request the audience to type their questions in the chat window, because we will take all the questions at the end of the sessions. We have 4 talks, and I would like to invite my first speaker, Professor Rodney Scott. He is working as a professor at the School of Biomedical Sciences and Pharmacy, College of Health, Medicine and Wellbeing, University of Newcastle, Australia. And he's very internationally recognized leader in genetics, and his work is around the inherited forms of colorectal and breast cancer genes, and he has published more than 500 scientific articles, and we very much look forward to his talk, and the title of his talk is The Changing and Challenging Natural History of Lynch Syndrome. Over to Professor Rodney, please.
Back again. I'm going to talk now about something quite different to breast cancer, the changing and challenging natural history of Lynch syndrome. Now, what do we know about Lynch syndrome? Well, we know that it's associated with one of four mismatch repair genes, MLH1, MSH2, MSH6, or PMS2. It confers a high risk of epithelial malignancy. The carrier frequency is estimated to be less than 1 in 300. Disease penetrance is based primarily on retrospective studies. Colorectal cancer is the most frequent disease in Lynch syndrome overall, but endometrial cancer is the most frequent disease in women. And the genetic phenotype is associated with something called microsatellite instability, and we'll come back to that in a minute. The often reported cancer risks in Lynch syndrome or hereditary non-polyposis colorectal cancer are shown here, with colorectal cancer being at about 80% by 80 years of age, endometrial cancer around 43 to 45% at 80 years of age, and stomach, biliary tract, urinary tract, and ovarian tumors presenting somewhere between 10 and 20% by 80 years of age as well. Now, one of the dogmas is that disease is believed to follow the adenoma-carcinoma sequence, and this was put forward many years ago, and this diagram here shows the adenoma carcinoma sequence that was proposed many years ago by Fearing and Vogelstein, as shown down here. And then once the DNA mismatch repair genes were identified, there was some debate as to when they became active and how they interacted with the development of carcinomas and frank cancers in individuals with Lynch syndrome. They certainly resulted in a different range of genes being affected. But nevertheless, the outcome was cancer. And of course, the thing that we want to try and avoid is, if we are lucky enough to develop cancer, is either local recurrence or metastatic disease, or both. Now, I mentioned DNA microsatellite instability. That can be measured, and that's shown in here, and it correlates with the staining of immunohistochemical markers that target MSH2, MLH1, PMS2, or MSH6, and that's shown, some of them are shown here in this diagram here. And what you're looking at, this is the normal expression of these markers, or the normal distribution of these markers, but in a tumor, you see that it's shifted. In this case, it's shifted more towards the left here and here as well. You may not necessarily see it in all markers, but it's occurring in some, and you're getting a bit of a shift in here as well. And similarly, you can see the same thing here. And the more markers you look at, the more confident you can be that you're actually looking at DNA microsatellite instability, and that is a hallmark of Lynch syndrome. Now, during the course of investigating Lynch syndrome, there were several questions that began to become prominent because certain discrepancies were being revealed by different groups around the world. And the prospective Lynch syndrome database was established to try and obtain better information on Lynch syndrome patients and compare what we collect prospectively against retrospectively cross-sectional data, so that we can get a better idea of which one's reflecting the true events that are occurring within the disease, or are they indeed both similar. This would then lead to a better knowledge base on the natural course of the disease and the effect of the interventions that were being offered to patients with Lynch syndrome. So what can we say is emerging out of the Lynch syndrome database today? Well, the first thing is that there doesn't seem to be much difference in survival between patients having annual colonoscopy compared to once every 3 years. The disease risk appears to be much less than originally estimated from retrospective data. And as more individuals are surviving longer, other malignancies are becoming more frequent. So if we look at the survival functions after colonoscopy, and we've broken it down here into stage 1, 2, 3, and 4 stages, we find that the survival function for stages 1 to 4 is relatively okay. This is the survival after colonoscopy, and this is for people who don't have disease. And over here we have the survival for stages 1, 2, and 3 disease, and stage 4. So we get survival by stage, and we see, as might be predicted, stage 4 disease doesn't do very well at all, but stages 1 to 3 do surprisingly well. This is also witnessed with respect to the frequency of colonoscopy, where we don't see any significant difference in the stage, whether you do a colonoscopy annually or once every 3 years or over once every 2.5 years. Now, if you were assuming that colonoscopy was going to reduce the staging of disease because you were removing premalignant tumors at earlier stages of disease, you'd expect it to change as you got more and more frequent colonoscopies. But that doesn't seem to be the case. So this is up here spread over 4 time points, and over here 2 time points, and we don't see any appreciable difference. What we also see is that the cumulative incidence by age is not quite the same as we originally thought, in that MLH1 and MSH2 for all cancers or any cancers is relatively high by 70 years of age, being somewhere around 70%. MSH6 is lower, just over 55%, and PMS2 is even lower still, just under 20%. And that's for any cancer. If we look for colorectal cancer, we find something quite interesting in that PMS2 doesn't seem to appear to be associated with colorectal cancer risk at all. MSH6 is associated with about 20% risk at 70 years of age. MSH2, more specifically, is associated with about a 35% risk, and MLH1 about a 45% risk. And that's for colorectal cancer. If you look at endometrial cancer, it's quite a different picture. PMS2 is associated with a risk that's hovering at around 25%, maybe a little bit less. MLH1 is just over 30%, and MSH6 and MSH2 account for 50% risk by 70 years of age. So it's highly intriguing that these genes appear to be associated with different facets of Lynch syndrome. Now, one of the dogmas which we've hinted at is colonoscopy reduces the incidence of colorectal cancer by removing the offending precancerous lesion. And these lesions are shown in this diagram here. If you don't do chromographic colonoscopies, can be very difficult to identify, especially a lesion like this, and they can be bypassed. However, if you do chromographic colonoscopy, you find that you can actually easily see where this adenoma is developing. The difficulty is in removing these adenomas and making sure that you've got all of them removed. And the question remains, do all colorectal cancers develop for an obvious lesion, even the flat adenoma? Now, we know that evidence-based data suggests that polyps, if they're removed, results in a significant decrease in adenoma development and consequently colorectal cancer. And this has been shown very well in the National Polyp Study on colorectal cancer mortality, where the general population, if it's offered colonoscopy, has a significantly lower rate of mortality compared to individuals who don't have any interventions. Nevertheless, some people still die of colorectal cancer, as indicated here, and they don't even have an adenoma. We need to know what's going on there. But what's this data telling us? The fact that increased colonoscopy does not appear to be associated with any reduction in the staging of colorectal cancer, nor does it appear to be associated with the frequency of disease. So, summarizing this, we can say that colonoscopy polypectomies are common, and therefore there should be a reduction in cancer diagnosis. We're talking specifically about Lynch syndrome here, who have annual colonoscopy compared to those who have it once every 3 years. We don't see that. Furthermore, we don't see that disease stage is influenced by colonoscopy. And there's an unusually long disease-free interval or survival that's not explained by the tumor markers themselves. The disease risks appear to be much less, and this is specifically for colorectal cancer, than expected, with virtually no difference in disease risk compared to the population data for PMS2. Now, you could argue that the quality of the colonoscopy plays a role in these findings because colonoscopists vary, bowel preparations are not always perfect, subadenomas are difficult to identify. But if you take all these three things together, they're unlikely to account for these findings, because if it's averaged over a number of colonoscopists, they're still going to see these differences. A better explanation is that there may be something biologically different in Lynch syndrome compared to the majority of colorectal cancer patients. Immunohistochemistry results from normal appearing colonic tissue from patients with Lynch syndrome may be telling us something about the nature of tumor development in this syndrome. So if we look at these diagrams here, we find that, for example, here are two colonic crypts. They stain for PMS2, and that's fine. But here's a crypt that doesn't. If we look here, we can look at the colon, we can see one crypt that stains for PMS2, and here are 4 that don't. Similarly, for MLH1, we can identify crypts that don't stain for MLH1, or even MSH2. And I missed the other one out for MSH6, but you see the same thing. This suggests that crypts are there are many more crypts that are deficient in DNA mismatch repair than you would otherwise expect in an individual with Lynch syndrome. So what do these findings imply? They imply that you should see many more malignancies than what is observed. Lynch syndrome patients should not survive as well as they do. The frequency of colorectal cancer should be much higher in younger patients than you'd otherwise expect. And other extracolonic cancer would be much less common, except potentially for endometrial cancer. And this would be simply due to the fact that they die of colorectal cancer and they don't survive. So what's an explanation for this? Well, one explanation is that DNA mismatch repair errors result in unusual peptides due to DNA polymerase slippage, and these are termed frameshift peptides. Frameshift peptides are recognized as foreign. Frameshift peptides are presented by the MHC as the major histocompatibility complex as non-self. Frameshift peptides presented by the cell are targeted and removed by immune surveillance, leading to clearance of malignant or pre-malignant clones. And you have to take into account that immune surveillance is not perfect, and some tumors do develop, be it at a frequency that's much less than expected. So the presence of frameshift peptides predicts better survival, as shown in this diagram here. So if your tumor is microsatellite unstable or instable, you'll develop frameshift peptides. Frameshift peptides are immunogenic, and you'll see a tumor lymphocyte infiltrate, which is reported to be quite common in Lynch syndrome. Whereas if you're microsatellite stable, you don't get the tumor lymphocyte infiltration, and you don't get the presentation of frameshift peptides, making these tumors much more dangerous than the ones that are associated with microsatellite instability. You can, of course, get some stable tumors converting to unstable ones, and we see that in about 15% of all colorectal cancers. So frameshift protein-specific T-cell immune responses are not present in individuals with MSS cancer or microsatellite stable cancer. They are present in individuals with microsatellite instability associated cancers. And if you don't have an MSI cancer, but you do have Lynch syndrome, you can still develop colorectal cancer, and you can also then convert to frameshift-specific peptides. And if you look at the overall survival of MLH1 and PMS2 individuals compared to years once they've been treated, it's significant. Okay, and you don't see that for colorectal cancers that arise on a background of, for example, APC mutations. It's got nothing to do with microsatellite instability. So what we have is microsatellite stable hosts, peripheral tolerance to proteins. If you have mutations in mismatch repair genes, you're likely to get a microsatellite instability high phenotype, which generates these novel frameshift polypeptides. There's no peripheral tolerance to frameshift peptides. The tumor is immunogenic to the host. You get the generation of an immune response, increased levels of tumor-infiltrating lymphocytes that are associated with MSI high tumors, fewer metastases, and enhanced survival. So, in summary, the PLSD, the prospective Lynch syndrome database is providing new insights into the natural history of this syndrome. The presence of normal appearing crypts that do not express given mismatch repair proteins support the definition of Lynch syndrome. There are 4 genes associated with this syndrome, and they behave quite differently. Disease survival is much better than expected. As patients age, different malignancies are being reported, and that's probably related to the fact that they are surviving their colorectal cancers due to better treatments over time. And frameshift peptides appear to influence disease risk. Deficient mismatch repair predicts efficacy of PD-1 and PD-L1 blockade, and that's indeed been shown to be the case. Thank you. If any questions, I'll be happy to answer them.
Thank you very much, Professor Rodney, for your talk, and it was very informative. And we will take the questions at the end of this session. So moving on to our next speaker, I would like to introduce to the audience who just joined us that Professor Dr. Jörg Heisel is working as a head of functional genome analysis, German Cancer Research Center, TKFC, Heidelberg, Germany. And he is working on pancreatic cancer. And the title of his talk is Toward the Understanding of Molecular Tumor Communication in Pancreatic Cancer. So over to Professor Dr. Jörg, please.
Hello, this is again Jörg Heisel with my second presentation of the work we are doing here in my group. What I will talk about is aspects of molecular communication within tumors, in particular pancreatic cancer, which is the focus of the kind of work we are doing. And starting my presentation. Just as a quick reminder, I'm working at the German Cancer Research Center in Heidelberg in Germany, which is a research institution of about 3,500 people working on all aspects of cancer research, and starting from basic analysis really to translational work, which has been done at this NCT facility that we run together with the university clinics in here Heidelberg, but other cities in Germany as well, where really the results of the research is going to be applied as quickly as sensibly possible to patients. The focus of my group is pancreatic cancer, as I forwarded to you before, and we do analyze all molecular kind of aspects of pancreatic cancer and try then to use that data to find functional variations that may be usable for early diagnosis, therapy, or other means, just basic understanding really also of the cancer as it is. For the simple reason, pancreatic cancer is one of the cancers for which basically there's not much of a treatment. Surgery is possible for about 20% of patients, but even they will eventually die of pancreatic cancer, since all of them basically have a relapse, usually within 2 years after surgery. So what is really needed is new targets, new processes for early diagnosis, and this is what we are trying to achieve. Obviously, a tumor tissue, as shown on the right, in our case, pancreatic ductal adenocarcinoma, is not just tumor cells, but actually consists of a lot of cells. And that makes quite a difference if you think about the biology, since we do not just need to understand the tumor cells, what is ongoing in tumor cells, what makes them different to the non-tumor cells, but we need to understand the entire tissue, which means all the other cells that are part of a tumor as well. And if you just look at how a pancreatic cancer looks like, a tissue looks like, what has usually been used to identify biomarker molecules are samples which contain more than 50% of tumor cells, which is useful if you really want to find markers dealing with the tumor rather than with the tumor tissue. But the average patient lies over here, which means the average patient, or the tissue, the pancreatic cancer of an average patient has between 5 and 15% of tumor cells, while 95 to 85% are something else. That means biomarkers that you identify with the high tumor samples may not actually work very well in the average patient. So what we looked also at, for instance, is if there are certain activities, which means genes or proteins that are particularly expressed in a particular kind of cell in the tumor tissue. And what you see here is really that, yes, it's true. There are actually certain genes, means proteins, which are specifically high or low expressed in particular cells within a cancer tissue. There's a clear correlation, as you can see on the left, for 4 of those genes. which is also true for the proteins. And in the table you see we have about 1,000 proteins which we know are typically expressed in a particular cell type within the tumor tissue. And as you can see on the graph on the right bottom, really, the overlap between them is really little, very little. It's two genes, proteins really overlapping between tumor cells and, well, normal cells, which is a kind of strange label, but anyway, and inflammation is yet again different. So which means just looking at the expression profiling, either at the RNA level or at the protein level, allows us really to make a molecular pathology, which means we can tell you just by the expression profiling what percentage of tumor cells in the tissue we looked at, what percent of stellate cells, pancreatic cells, and so on and so forth, which means we do have this information and could do a kind of molecular pathology, which is not the eventual objective of this kind of work, but it's interesting that this is actually possible. That means, of course, if particular cells have particular genes active or non-active, it's more likely, and it's known, of course, that the cells within a tumor tissue interact with each other and affect each other, which means stellate cells, once they get activated, which is what they are in a tumor, actually affect, for instance, the tumor cells, triggering growth and proliferation of tumor cells, or for instance, macrophages, M2 macrophages in case of pancreatic cancer, actually trigger metastasis and invasion behavior of the tumor cells. And obviously also the tumor cells have an effect on these cells and others too. And this is what we're actually looking at, really looking at the interaction of the various cells on all the other cells of a given tissue, which is in our case pancreatic cancer tissue. For that purpose, we actually built a particular kind of model system, if I may say. It's a multi-well plate, but the difference is actually that the wells, as shown here schematically, are not necessarily separated in A, B, C, D, which means 4 different cell types, for instance, but actually they have a common room and share this room, which means via this room, while the cells are remaining in the location in A, B, C, and C, actually what they are releasing into the growth medium is being transferred to all the others, which means the interaction can occur. And as a matter of fact, by sealing 1 or 2 of these chambers for a particular time or during a particular period, we can actually kind of control this exchange of molecular information between the different cell types. A simple sample shown on the left bottom, there are tumor cells that were grown with gemcitabine, which is a drug that inhibits proliferation of cells. And the same experiment was done also, but in presence of pancreatic stellate cells, activated pancreatic stellate cells. And what you see on the right is really then the result. The tumor cells grown alone with gemcitabine, the drug, there's a reduction to about 50%, or by about 50% in growth of these tumor cells, while the same cells are grown under the same conditions, but in presence, of what the pancreatic stellate cells release into the medium coming across from here to there. As a matter of fact, the growth of the tumor cells was very little affected. The effect on the tumor cells of gemcitabine was rather little, insignificant as a matter of fact. We also did much more complex analysis, one of which was, for instance, to find out what triggers the development of M2 macrophages, or, the point is monocytes, so immune cells, that are circulating in the blood upon there's a pancreatic cancer, they develop and they develop into particular subtypes. For instance, M1 macrophages are an indication of good prognosis in case of stomach cancer, colon cancers, and others. While M2, for instance, are an indication of a very bad prognosis, particularly for glioma and breast and pancreatic cancer. So what we were interested in was, what effect do activated pancreatic stellate cells, activated by TNF-alpha, which is released actually by the tumor cells, and are they secreting, and what does this secreted, or these secreted proteins, what effect do they have on the development of monocytes? Will monocytes develop? And if they do develop, will they develop into M1 or M2 macrophages, as a matter of fact? And the result you can see here. So this is flow sorting analysis. What you see as an example, typical example for an M1 macrophage, here a typical example for an M2. And this is a result we obtained of macrophages, monocytes grown in presence of the release of the secretome of TNF activated pancreatic stellate cells. And you can see, they really indicate very clearly M2 subtype. And the same is true with the secretome of tumor cells. It's again M2. As a matter of fact, what you see, all the yellow ones here, this is the control, and these are the ones we actually looked at, have a strong M2 phenotype, phenotype by flow sorting, where, for instance, if the secretome has been used from non-activated pancreatic stellate cells, I mean, the cells, actually, the monocytes develop into M1 type of macrophages. The same can also be seen here in terms of cell morphology. This is M1 macrophages, this is M2 macrophages. And this is monocytes that had developed in presence of the secretome of activated pancreatic stellate cells. And simply by the morphology, you can see, apart from this, clearly, that they are M2 type mostly and not M1 type. What we also did is, as a matter of fact, we mixed all the cells shown on the left bottom here, so tumor cells, actually stems-like tumor cells as well, then tumor-associated fibroblasts, M2 macrophages, and activated stellate cells, and grew them put together, and then generated a methylation profile on 450,000, which is, I think, about 3% of all the methylation sites in the human genome. What you see is a profile to the left of healthy tissues. This is really entire tissues. So the profile obtained on them. On the right of tumor tissues, pancreatic cancer tumor tissues. And this one line here really is the one that indicates the mixture of all these 5 different cell types. And what you can see at the molecular level in terms of methylation, but the same is true also for other expression analysis, for instance, actually this mixture of cells very much resembles what you see in the profile of a tissue, which means the mixture of cells actually resembles closely a tissue, which means it could act actually as a model of the tissue, which is the direction we are moving to actually work out, what may help in terms of treating pancreatic cancer better than it is possible at the moment. What you also see is, for instance, that quite a few genes are highly methylated and highly methylated even in their promoter, which is kind of interesting because quite a few of them are actually active nevertheless. And usually the standard model is, of course, once a gene gets methylated in its promoter, it's been shut down. But as a matter of fact, we did this kind of analysis. What you see here, this is what you expect to see, which means high methylation. So on the right, high methylation, low methylation over here, high methylation. And that means on the y-axis, which is the transcript level, very little, relatively speaking, little transcript activity. But there's the same number of molecules of transcripts actually where high methylation of the promoter correlates with high expression of the gene, which means the gene is not actually shut down, but on the contrary, in many cases, actually it's triggered and activated upon methylation of its promoter sequence. And we looked at that in more detail, and we did this by using an array that contained about 700 human transcription factors, and then incubated with promoter sequences, methylated or unmethylated. And what you see here, and I just highlighted a few examples, certain transcription factors actually bind specifically to the methylated DNA. They do not bind to the non-methylated DNA. Other transcription factors actually bind specifically to the non-methylated DNA, but not to the methylated DNA. And various transcription factors don't care whether the DNA has been methylated or not. They just bind anyway in both cases. And that means, obviously there are cases where methylation is being used to activate a gene rather than to shut it down. We looked into this, and this has recently been published, so I'm not going into much detail, but looking at certain pathways and so on and so forth, for instance, finding this particular transcription factor to bind to the promoter of this particular gene, which is associated to cancer development closely. It doesn't bind to the unmethylated sequence, it does bind to the methylated sequence, kind of penetrating this the complex structure of the DNA. Nevertheless, although it's been highly complex and covered by all other proteins, nevertheless this is able to bind to the methylated promoter sequence. Then it's recruiting cofactors, and based on that, then the highly methylated gene or promoter is highly methylated. It's getting activated and transcribed. What is also interesting, though, particularly these genes could actually work quite nicely, or the methylation of these promoters, for diagnosis of pancreatic cancer, since this also seems to happen in some cases in normal tissue, but more specifically in cancer tissue as it looks. This is kind of interpretation of the data that we have. If you look at cluster analysis on profiling data, so DNA methylation, mRNA transcription profiling, microRNA, or combinations thereof, you can see, well, you can distinctly nicely separate healthy tissue from diseased tissue. We cannot discriminate between chronic pancreatitis, which is an inflammation of the pancreas, and actual tumor of the pancreas by normal clustering. But using machine learning, we are actually able, and here's a process, it's rather complex, took a lot of time to actually get it to work and to classify the appropriate molecules. But in the end, what we ended up with was simply 6 CPG sites, so 6 methylation sites in a particular gene that actually allowed a distinction between pancreatic cancer and the inflammation of the pancreas, chronic pancreatitis. And this is the most challenging thing, because in clinical reality, what is happening that this tumor and the inflammation of the pancreas cannot be discriminated easily. Imaging systems don't work usually, or pretty badly. Even the molecular analysis doesn't work that well so far. And that means that only 2/3 of the patients get diagnosed correctly, and 1/3 of the patients get diagnosed the wrong way, which means a chronic pancreatitis patient is diagnosed to be cancer and may undergo surgery, which is unnecessary since there is no tumor to be removed. But on the other hand, cancer patients may be diagnosed to have chronic pancreatitis, and the treatment of the tumor, which is urgent to have any effect in the first place, is delayed because of this false diagnosis, which means having 1/3 of false diagnosis is pretty bad. And we hope and think that by this process here, with this methylation data and the ability to really clearly discriminate absolutely between chronic pancreatitis and cancer, we will improve that situation significantly and thereby improve survival, at least survival time of pancreatic cancer patients in particular, quite significantly. With this, I'm done again. Wish to acknowledge, though, the work of Andrea, who organizes all the work on pancreatic cancer in our group, and particularly Yenan Wu, who worked on the methylation data I've shown you before. So these are the 2 people who did most of the work, what I've just shown. And with this, I thank you very much for your attention, and I would be happy, really, to answer any questions you may have. Thank you very much.
Thank you, Dr. Yong. That was very, very nice talk. And I have many questions myself. And I can see that there are questions appearing in the chat window here as well for you, which we will take at the end of the talks. So with this, I would like to introduce my next speaker, Dr. Marcela Hill. He is the director, Center for Translational Immunology Institute, Pastor de Montevideo at Uruguay. And his main interests are in immunoregulation and immunotherapy. Earlier in the first session of the cancer research in the symposium, he shared a very interesting work with us. And now he is going to talk on unleashing inflammasomes to improve immune checkpoint blockers therapy. Over to Dr. Marcelo, please.
Hello, welcome to this presentation where we are going to talk about one of the last revolutions in oncology, which deals with the immune checkpoint blockers therapy. And we are going to focus on how we can improve this therapy by modulating inflammation, and particularly by modulating inflammasomes activation. So, as you know, anti-CTLA-4 and anti-PD-1 therapies have increased overall survival and progression-free survival in several cancers. However, most of the patients do not benefit from these therapies because they have no clinical responses. So we need to identify primary and secondary resistance mechanisms to be targeted and improve the anti-tumor efficacy of these approaches. We also need biomarkers to predict clinical responses, and in part also because these treatments are very expensive and health systems need to rationalize the expense in these approaches. And one of the main questions in this field is which is the role played by inflammatory pathways in determining the clinical outcome. And that's why we are focusing today on a potential for inflammasome activation in improving the anti-tumoral efficacy of anti-PD-1 and anti-CTLA-4 therapies. So we need to improve the anti-tumor efficacy. As I said before, most of the patients do not benefit from these approaches, even if we combine anti-PD-1 and anti-CTLA-4. There is also a huge problem with toxicity with immune-related adverse events, which are triggered by these therapies, and these problems can lead to stop the medication or even the death of the patient. And as I said before also, we need to predict these clinical responses. So how can we answer all these questions? I think that the key point is that we need to understand the mechanisms, and there is much to do in this field. And it is thought that understanding this mechanistic basis will lead to a better management at the clinical level. So how do these approaches work? So in normal T-cell responses, you have that T-cells are recognizing antigen in antigen-presenting cells, mainly dendritic cells for naive T-cells. So you have antigen recognition through presentation by MHC molecules, and this is the so-called signal 1. But in order to activate these T cells, you also need the so-called signal 2, which is provided by costimulatory molecules such as B7, CD80, and CD86, which will ligand CD28, and this will lead to the full activation of the T cell, leading to cytokine release. However, there is a physiological break for this activation, which is manipulated in cancer by malignant cells. So in the setting of in the tumor microenvironment and in the tumor draining lymph nodes, when T cells are recognizing the antigen, you have the costimulatory signals, but you also have co-inhibitory signals such as PD-1 and CTLA-4, and in this case you have no T cell activation and you have no cytokine release. So when you block either PD-1 or the ligands PD-L1 and PD-L2, or this is true also for anti-CTLA-4 antibodies, you can block these inhibitors, and then in this scenario you will have cytokine release and you will have a strong anti-tumoral immune response. So the dogma says that hot tumors, inflamed tumors, have a high probability of clinical response, whereas non-inflamed or cold tumors, because the inflammatory infiltrate is excluded from the tumor, or whether there is no infiltrate at all, in these cold tumors you have a very low probability of response to immune checkpoint blockers. And this is the main idea. This, as I said before, is a kind of dogma. Accordingly, we know that there are different inflammatory cytokines which are associated with the anti-tumoral effects of immune checkpoint blockade, and this is true for anti-PD-1, anti-PD-L1 antibodies, and also for anti-CTLA-4. However, there is also several papers showing that, at least in some scenarios, pro-inflammatory cytokines can be associated with bad response to immune checkpoint blockers. So you have immunoregulatory cytokines such as TGF-beta, but it is also true that strong inflammatory cytokines such as IL-1 beta, TNF-alpha can also lead to a bad outcome when blocking PD-1 and CTLA-4 in cancer. So, which is the role of inflammation in immune checkpoint blocker outcome is really a big question. Still, we do not understand completely how inflammatory mediators, and particularly inflammatory cytokines, are determining the clinical outcome of immune checkpoint blockers. So we are working on inflammasomes, which are a specific platform in the cytosol, which is activated once cells are stressed because of different stimuli. So inflammasomes are cytosolic complexes that regulate the activation of the inflammatory caspase-1, and then caspase-1 can lead to pyroptotic cell death or secretion of inflammatory cytokines such as IL-1 beta as well as IL-18. So we wonder whether inflammasomes could be regulating the antitumoral immune responses triggered by immune checkpoint blockers. So we published this paper in 2019, and here we describe TORID or TMEM176B as a key inhibitor of inflammasome activation. And we observed that inflammasomes are promoting antitumoral immune responses, particularly in the context of anti-PD-1 therapy. And in this paper, we describe the compound BK as a TMEM176B inhibitor. So BK is improving the antitumoral efficacy of anti-PD-1 antibodies in an inflammasome-dependent manner. So one of our first questions was whether inflammasome could play a role in triggering antitumoral immune responses in the context of immune checkpoint blockade therapy. So here we worked with wild type and caspase-1 knockout mice, and we observed that there was a significant decrease in the antitumoral efficacy of anti-CTLA-4 and anti-PD-1 therapies in the inflammasome knockouts caspase-1 knockout mice. So you can see here that the overall survival of the mice was diminished in the inflammasome knockout mice as compared to the wild-type animals, and a similar result was observed with the knockout of one of the components of the inflammasomes, which is the sensor NLRP3. So inflammasomes do seem to play a role in antitumoral immune responses triggered by anti-CTLA-4 and anti-PD-1 therapies. So this role of inflammasomes may play a role at the clinical level, and here we analyzed a signature of 15 inflammasome-related genes, and we analyzed melanoma patients being treated with anti-PD-1 therapy, and we analyzed responder and progressor patients. We observed that in 5 out of 5 responder patients, this inflammasome signature was increased during the treatment with anti-PD-1 therapy. In contrast, in progressor patients, we observed that in 4 out of 7 there was a decrease in this inflammasome signature during the treatment as compared to the pre-treatment stage. In that work, we also described BK as a TMEM176B inhibitor. So TMEM176B is an inflammasome inhibitor. So the idea here is that with BK, we are unleashing inflammasome activation. And when we treated tumor-bearing mice with BK, we observed that in the tumor-draining lymph node, we observed increased inflammasome activation. Here we are measuring caspase-1 activation in vehicle or BK-treated animals, and we observed increased inflammasome activation in the tumor-draining lymph node of the animals being treated with BK. When we analyzed the survival of these mice, we significantly protected them from death when treated these animals with BK. And this effect was inflammasome dependent, since we completely lost efficacy in inflammasome knockout animals such as caspase-1 deleted mice. I'm not showing here for the sake of time, but this effect triggered by BKs was completely dependent on CD8 T cell responses. If we delete CD8 T cells, then we again completely lose the efficacy of this BK therapy. We then combined anti-PD-1 or anti-CTLA-4 antibodies with BK, and you can see here in a lymphoma model that when combining here in the red line BK and anti-CTLA-4, we significantly improved the efficacy of the monotherapies, and this was true also when combining BK with anti-PD-1 in lymphoma, melanoma, colon and lung cancers. We improved the antitumor efficacy of immune checkpoint blockers by adding BK to this therapy. So again, unleashing inflammasome activation is improving the efficacy of immune checkpoint blockers. But there is this PNAS paper which shows completely different results. In this paper, Abt and colleagues have shown that blocking IL-1 beta improves the antitumoral efficacy of anti-PD-1 therapy. So you can see here that when you have here the tumor growth in anti-PD-1 treated mice, and then when combining blockade of IL-1 beta, you significantly improve this effect. So these results are somehow the opposite observation, right? When you block an inflammasome-related cytokine such as IL-1 beta, you are improving the antitumoral efficacy of immune checkpoint blockers, in this case anti-PD-1. So one of the differences with our work is that this is done in a breast cancer model in mice, but we thought that although this is a difference, that there may be other explanation for this opposite result. And one of the interesting things that we found is that in this paper, the protocol was, in the timing, was really opposite to our protocol. In this paper, researchers have performed first IL-1 beta blockade and then PD-1 blockade. Whereas in our research, we always did the first time anti-PD-1 and then inflammasome unleashing with BK. And if we do the other way around, that means if we come first with BK and then with anti-PD-1, we completely lost efficacy. So we think that timing does matter, and we think that timing would be an explanation of these different observations. So to understand how timing could play a role, we think that again we have to focus on mechanisms, and you know, T-cell exhaustion is one of the main mechanisms by which anti-PD-1 therapy is modulating antitumoral immune responses. So what is T-cell exhaustion? So exhausted T cells are a distinct cell lineage that arises during chronic infections and cancer in mice and humans. So exhaustion is a kind of T-cell dysfunction, and the main hallmark is that you have a progressive loss of effector function and proliferative potential in exhausted T cells. Together with those functional issues, you have sustained and high expression of inhibitory receptors such as CTLA-4 and PD-1, and this is complemented with altered metabolisms, a particular subset of transcription factors such as TOX and TCF1, and a particular epigenetic problem. So the idea is that in normal immune responses, most of your T cells will differentiate either into effector T cells or into memory T cells. However, in situations such as chronic infection and cancer, your T cells will not be differentiating into effectors but into exhausted T cells. And importantly, within the exhausted T cell subset, T cells, you have two main subsets. You have early exhaustion, where you have expression of TCF1, and these cells have the possibility to proliferate when you block anti-PD-1. And these cells can also differentiate in an inflammatory context to terminally exhausted T cells, where they can still kill malignant cells through Granzyme B dependent mechanisms. So these progenitor exhausted T cells, which express the transcription factor TCF1, you can expand these cells with anti-PD-1 therapies. And then these cells can be induced to differentiation to Granzyme B positive cells. So these exhausted T cells, terminally exhausted T cells, have the ability to still kill malignant cells, although this capacity is diminished when compared to effector T cells. But when you block PD-1 with anti-PD-1 antibodies, you are inducing the expansion of these T cells, progenitor exhausted T cells, which can then differentiate into these terminally exhausted T cells. So we think that the timing, this timing issue could have a mechanistic base in these subsets of exhausted T cells. So the idea here is that you perform first anti-PD-1 therapy. You are expanding these precursor exhausted CD8 T cells, and then if you activate the inflammasome, you may be inducing the differentiation of these T cells into the terminally exhausted that have the capacity to control tumor growth. However, if you do this protocol the other way around, first inflammasome activation therapy, you will be inducing the differentiation, but without expanding these cells in anti-PD-1 therapy, because these cells do not have the capacity to respond to anti-PD-1 blocking therapies. So we have experimental data which support this hypothesis. So here we are analyzing different subset of exhausted T cells within the tumor microenvironment. So we analyze progenitor exhausted, transitory exhausted, and terminally exhausted T cells. And these transitory exhausted are the cells that, although they are exhausted, they have some capacity to kill malignant cells. So if we compare the anti-PD-1 with BK, which here we call it boritinib, but here we are adding to anti-PD-1 the compound that unleashes inflammasome activation, you can see that we significantly inhibit progenitor exhausted T cells, and we think that this is because we are inducing differentiation of these transitory exhausted T cells that have some capacity to kill malignant cells. And these mechanisms are depending on Th17 responses, and one of the arguments to support this fact is that you lose this effect of unleashing inflammasome therapy when you analyze IL-17 knockout mice. So this capacity that have inflammasome unleashing therapy to diminish progenitor exhausted and to increase transitory exhausted T cells is completely blocked in IL-17 knockout mice. So to conclude, we think that different subsets of exhausted T cells play different roles in tumor immunity triggered by PD-1 blockade. Inflammatory cytokines have been shown to play contradictory roles in cancer therapy. So there are ambiguous roles of inflammatory cytokines in cancer immunotherapy. So we think that the timing when you combine these therapies is an important role, and this could be due to the modulation of different subsets of exhausted T cells. And we think that an axis of conventional type 2 dendritic cells and Th17 cells are differently modulating T cell exhaustion in the context of PD-1 blockade. So I would like to thank our collaborators in France, Cristina Couturi and Ignacio Anegon, in Argentina, the Gabriel Rabinovich groups, and in our team, Mercedes Segovia, Sofia Russo, which led this work on inflammasome activation and immune checkpoint blockade therapy. So thank you very much for your attention.
Thank you so much, Dr. Marcelo. It was very interesting talk. And there are some questions for you, which I will take at the end of the last talk. And I would like to introduce our last speaker, Dr. Rob Ewing. He is the director of the Graduate School of Biological Sciences at the University of Southampton, UK. And his main interests are protein-to-protein interactions with context of WNT signaling pathway. And we look forward to his talk. And the title of his talk is Multiomics Analysis of Protein Networks for Discovery and Development of Therapeutics. Over to Dr. Rob, please.
Well, thanks again for the invitation to present in the symposium. In this talk, I'd like to turn our attention towards so-called multi-omic analyses, where we use multiple types of omic analysis to investigate systems in cancer. And what I'll be telling you about is some of our work using multi-omic techniques to characterize protein networks, both for fundamental discovery, but also for potentially development of new therapeutics. So I start with this very high-level slide, and really, I think, state what everybody realizes is that disease states in humans are system-wide. And what I mean by that really is that when you think about the molecular networks that are perturbed in human disease states, such as cancer, then those molecular networks are really perturbed at many different levels. They're obviously perturbed at the genomic level, where we might accumulate, for example, somatic mutations in a tumor that result, for example, in altered gene expression. There may be altered methylation patterns, or, for example, changes in protein structure, function, and expression. A few years ago, the Cancer Genome Atlas really it's really a pioneering project for multi-omic approaches to cancer. And the Cancer Genome Atlas really has characterized thousands of tumors in terms of their molecular profiles at different levels, and is really now a fantastic resource for cancer researchers who want to do systems-wide analysis in cancer. But the point really here is that actually, integrated multi-omic analyses, and I'll be telling you about some of the work we've been doing in this area, I think are really the way forward here. If we want to fully understand these molecular networks, we have to begin to characterize them at all of the levels on which they operate. So what I'm going to do in this presentation is really provide you two case studies. I'm going to tell you about our work in two really quite different areas. The first is in talking about some of the work that we've been doing in the area of Wnt signaling in colorectal cancer, where we're using integrated multi-omic approaches to understand signaling networks. And the second case study I'll be telling you about is something very different, which is multi-omic approaches to understanding potential oncolytic therapy. So that is using a virus as an oncolytic agent in brain tumors. And I'll be telling you about how we're investigating how the Zika virus works in brain tumor cells using multi-omic approaches. What I'd like us to do, though, throughout the presentation is to really keep two questions in mind. The first is really a methodological question, is actually how can we do these kinds of studies? How can we take quantitative multi-omics data and integrate those and actually get better insights into disease? And the second question really to reflect on, I think, is what value do multi-omic approaches have in cancer research? And hopefully by the end of the presentation, I'll be able to provide you with some answers to these questions. So first off, I want to tell you about some work we've been doing in the Wnt signaling pathway. As many people will know, Wnt signaling is a pathway that is really intrinsic to many, particularly many types of solid tumor, and is typically associated with oncogenesis. So, for example, beta-catenin is an oncoprotein that functions in the Wnt signaling pathway, and you'll often find elevated beta-catenin expression or mutations in beta-catenin that allow it to accumulate in the cells in many types of cancer. But I just want on this slide to convey the idea actually that when we think about signaling pathways such as the Wnt signaling pathway, we need to think of them in terms of rather more complex networks. So, for example, in Wnt signaling, we know that there are multiple types of Wnt signaling. There's so-called canonical Wnt signaling, which involves beta-catenin, and beta-catenin is the main effector of the canonical Wnt signaling pathway. There's non-canonical Wnt signaling, which doesn't involve beta-catenin, which plays different roles in the cell, different roles in development, and different roles in cancer, and has different outputs. And just recently, actually, there was another pathway discovered, which is also beta-catenin independent, called Wnt stop signaling. So I think the overall message here, without going into all of the details, is that when we think about signaling pathways, we must think about them as networks, as rather complex networks, because these networks often or typically have crosstalk with other signaling pathways, for example. We know that there's an interplay between canonical and non-canonical Wnt signaling. And we need to move away from just considering the outputs, really, of signaling pathways, such as the Wnt signaling pathway, as being purely transcriptional. So there are many different types and layers of output that result from oncogenic activation of signaling pathways that we need to understand if we're to understand these pathways in a systems manner. So this really argues for a multi-omic approach. We want to study pathways such as the Wnt signaling pathway. We need to take multi-omic approaches where we're analyzing the networks at multiple levels. Now, the case study I'm going to use to illustrate some of our approaches is some work that we did a couple of years ago with the oncogenic beta-catenin signaling pathway. So remember that beta-catenin is this oncoprotein that accumulates in the cancer cell, typically, and very often the mechanism behind that accumulation is the accumulation of mutations in beta-catenin itself. And what I'm going to talk about really are some of the most well-studied mutations. These are mutations that actually take away the ability of the cell to control the levels of beta-catenin. So what actually happens, you get in-frame deletions in beta-catenin, which remove codons that would normally code for serine residues in the protein. And those serine residues are normally phosphorylated, and that leads to subsequent degradation through the proteasome. And so by removing the serine residues, these mutations allow the protein to accumulate. And this has been actually observed particularly in colorectal cancer, but in other cancers as well, as an oncogenic mechanism. So a few years ago, we took colorectal cells harboring these mutations and did this experiment. And I'm really just going to present this as kind of a case study for multi-omic analysis. So what we did, we started out with parent cells that had both the wild type, the wild type in green here, and the mutant beta-catenin in red. So it's a heterozygous mutation. And then we used, in addition to those parent cells, we used mutant cells which had only the mutant allele, so that's the stabilized beta-catenin, and wild type cells that had only the wild type allele of beta-catenin, so no mutant protein. And what we did with these cells, we grew them up, and we used 3 types of omics analysis to profile the mutant and the wild type cells. So we use interaction proteomics, and this is where we take an antibody and we pull out the mutant or wild type protein and use mass spectrometry to characterize the interaction partners. We used expression proteomics, which is protein profiling. So we take the cells and extract the proteins and then measure the abundance of as many of those proteins as we can. And then we also used RNA-seq to characterize the transcriptomes of these two kinds of cells. And our objective in this analysis was to take all of this data from these experiments and combine it into meaningful biological networks that would tell us about the activity of the mutant or the wild type proteins. Now, I won't go into too much detail about the technicalities of how we do this, but in essence, you can think of these different types of data as layers. So, for example, we've got the transcriptome layer, we've got the expression proteome layer, we've got the interaction proteome layer, and essentially the kind of computational analysis is to take each of those layers and superimpose them onto known networks, and then to identify in those networks the areas of the network that are most perturbed across the different layers. So what we would do, for example, is to take large-scale protein networks from sources like BioGrid or HPRD, integrate all of our data, use a community, what's called a community detection algorithm, to identify subnetworks, and then identify these modules. And what this allows you to do is really collapse all of this different information across these different layers of the network into a single network that you can then analyze. So how do we actually analyze these data? Once we've put all this data together, we get these very large networks. From our beta-catenin experiment, we had a network of over 2,500 nodes with 10,000 edges in it. These we kind of think of as hairballs because they're very complex and they're actually difficult to really parse and understand. And so we use typically two kind of routes for interpreting these large-scale networks. The first is kind of like manual curation. So you take the known modules within the network and you can curate those and actually use your knowledge of the system to pull out interesting findings. So we do a lot of that kind of manual curation of these networks to identify functional modules. The other is a more automated approach, and what we do here is we use a community detection approach. So this is really identifying subnetworks within this larger network that actually identify particular targets. And I'm going to tell you in the next few slides about one of these targets that we followed up on. This is a transcription factor called ELF3, E74 like ETS transcription factor. And I'll tell you about how we identified this using the automated approach from this large integrated network. So one of the things that we do with these large-scale networks in our subnetwork identification method is to take that larger network and fragment it into different subnetworks. And essentially these are high-scoring networks where there's a lot of perturbation and where all of the nodes are actually quite well connected. And the approach that we used in this particular study was to take each of those high-scoring networks that we get from the large-scale network and ask, what are the transcription factors that actually are enriched with respect to these subnetworks? And in this case, we actually found something rather interesting. We found that the transcription factor ELF3, that I'll tell you a little bit more about in a moment, was actually scoring very highly with respect to many of the high-scoring networks. And so this gave us the clue that actually one of the drivers for some of the patterns that we were seeing across these networks was this transcription factor ELF3. So we're very keen to follow up on this particular protein. Now, just another methodological detail. Of course, what you could do is take the whole network and you could look at the enriched transcription factors. And if you do that, you see there are lots of transcription factors you identify. You can rank them according to the p-value, and you can see actually the one we got interested in, ELF3, was ranked about 16th in this list, and you know, it's one of those things you might see that finding and say, well, yeah, okay, that's kind of interesting, but it's not really compelling. But if you take the subnetwork-based transcription factors, where you essentially decompose that larger network into multiple subnetworks and then analyze those individual subnetworks, you can see the ranking changes. And actually, this was one of the reasons why we followed up with L3. We saw a very high ranking across multiple subnetworks for this transcription factor. So, of course, you know, that's not the end of the story, and you know, from these large-scale studies, if you identify a protein of interest, you always want to sort of follow up and validate that. And we were, in collaboration with Yuhua Wang and his group, we did some follow-up experiments, and those, in essence, those follow-up experiments confirmed to us actually that ELF3 is an antagonist of Wnt signaling and also of EMT. So, for example, we looked at some of the markers for EMT, such as ZEB1, and found that actually if you repress ELF3, this is in a different model, but if you repress ELF3, you get elevated ZEB1 activation, and that's associated with EMT. We also found that there seems to be a mutually repressive relationship between the effector of Wnt signaling, which is beta-catenin, and ELF3. And our sort of underlying hypothesis here is that actually ELF3 has to be overcome because it's a repressor of EMT by beta-catenin. Okay, so beta-catenin has to overcome ELF3 activity in order for EMT in these epithelial cells to proceed and epithelial cells to be transformed into mesenchymal cells. We also followed up with some large-scale analyses. So I told you about the Cancer Genome Atlas, TCGA, earlier in the presentation, and TCGA is a really powerful data set for validating some of these findings. So down here in this panel, what you're looking at here are, these are colorectal tumors, 270 colorectal tumors taken from TCGA. And the patterns you're looking at here, these are beta-catenin associated patterns. So these are genes for which the expression is correlated with beta-catenin, so that Wnt effector. And these are the ones that are correlated with ELF3. And you can see even by eye here that there are mutually antagonistic or exclusive expression patterns for the ELF3 correlated genes and the beta-catenin correlated genes. So you can see these are kind of mutually antagonistic. The other thing I'll point out here is we did some analysis of colorectal adenocarcinomas themselves, and we found that actually there's a strong association of elevated ELF3 expression in tumours for which there was a better prognosis, and you can see that from the survival curve here, where you've got high ELF3 associated with a slightly elevated time of survival as compared to reduced ELF3. And we think this is actually related to the role of ELF3 as being a repressor of EMT and oncogenic Wnt signaling. So I've told you about some of that work with respect to Wnt signaling. I'd like to just use the last few minutes to tell you about a study which is really just ongoing. We've only really just started, but I think hopefully it will illustrate our multi-omic approach in a completely different area. I'm going to tell you about some work we're doing to understand whether the Zika virus can be used as an oncolytic agent against brain tumours. So many of you will be aware that in 2015-2016 there was a Zika virus epidemic in South America, particularly in Brazil. Of course, Zika is a mosquito-borne flavivirus. And one of the things that came out of that unfortunate epidemic is that the Zika virus during infection can cause severe, Zika virus infection during pregnancy can cause severe CNS developmental defects. So there were many kids being born with microcephaly, which is a birth defect that affects the development of the brain, to women who'd been infected with Zika in a particular window of their pregnancies. And what it was found, what came out of this, remarkable finding was that the Zika virus is capable of infecting neural progenitor cells, so NPCs, and this causes cell death in the developing embryo. Now, one of the unfortunately, through that epidemic, there were many kids born with microcephaly. But one of the bright sides that actually came out of all that was the finding by one of my collaborators, Professor Keith Okamoto, in Sao Paulo, Brazil. And Keith and his lab demonstrated that Zika actually can kill very effectively brain tumor cells. So not only does it affect neural precursor cells, which are the stem cells in the brain, it can very, very selectively kill brain tumor cells as well. I won't go through all the data here, but essentially if you infect these cells with Zika, they are very rapidly and effectively killed at very low multiplicity of infection with Zika. And so the thinking is that perhaps the Zika virus could become an oncolytic therapy against certain types of brain tumor. And I just want to mention in the last couple of minutes the work we've been doing in this area. So we've been working in conjunction with Keith Okamoto's lab. This is the PhD student Matthew Sherwood, who's working on this particular project. And we're taking a multi-omic approach to understanding how Zika infects these brain tumor cells. So one of the studies that we've been doing is to take the different non-structural proteins of Zika, express those in brain tumor cells, and then use mass spectrometry to analyze which proteins are linked to or associated with the Zika proteins to try and find out the interaction partners. And we think that's going to be a good way of really understanding how Zika causes the oncolytic effect that it does in brain tumor cells. And we're also using I said it's multi-omics, it is multi-omics. We're not just using proteomics, we're using RNA-seq, for example, to do transcriptomic analysis. So what we did in this experiment was to take the Zika viral infected brain tumor cells and also the neural precursor cells, infect them with Zika virus, and then to look across a time course following infection, and to characterize the transcriptome at each of those levels, at each of those points across the time course, to find out the dynamics of the process. And we'll be integrating that data with our interaction data that I just told you about to build up the networks, the viral interaction networks that actually cause the oncolytic effect. And we hope that what will come out of this will be identification of mechanisms that can either be used in future to target brain tumors, or to create therapeutics that can target the same mechanisms. Brain tumors, as I think many of you will know, in general are very poorly served by therapeutics at present, and we'd like to be able to add a new therapy mechanism to treat those particular conditions. So I'm going to finish here. Hopefully that I've managed to answer these questions, both in terms of the methodology of how we can use multi-omics data and how we can integrate it, but also the value of doing these kind of multi-omic approaches in different areas of cancer research. So I'd like to just finish by thanking some really key colleagues, great colleagues that I've had the pleasure to work with over the years. In particular, Mehmet Koyaturk, who's a computer scientist, who's really helped with a lot of the network analyses. Yiwa Wang, University of Southampton, who's really helped with teasing out some of these interactions and signaling pathways. Keith Okamoto, who's my collaborator in Brazil, for all his work on the Zika virus, and Hyong-Won Choi, who really has helped out in thinking about how to integrate multi-omic data. And I'd like to thank you also for your attention.
Thank you, Dr. Rob. We really enjoyed your talk, and it was very useful and highly informative. And I welcome all my speakers again, and very good to see you. And I would like to say that all the talks from all the speakers were really, really marvelous. And, you know, our participants were texting me that, you know, these are really good speakers that you have selected this time. So I would like to congratulate you on your good work as well. So with the short time that we have, I would like to take few questions, and I would like to take the first question from Dr. Marcelo. Dr. Marcelo, it's about CAR T cells, really. The question says that engineered CAR T cells change and may stop working after some time. In this context, how important it is to keep working in finding immunotherapies?
Okay, so yes, I think that the main reason to continue research on immunotherapy is that they have been a huge revolution in cancer treatment, right? So several advanced diseases that could be controlled in the clinic due to immunotherapy approaches by immune checkpoint blockers, and also by mostly in hematological cancer for CAR T cells. So I think that there is much research which is being done with other new checkpoints, you know, such as LAG3, TIGIT. So and some early clinical data show that these new checkpoints can add some therapeutic benefit over anti-PD-1, anti-CTLA-4, and I think that there are other important issues in immunotherapy research, for instance, which is the role of myeloid cells, and among these cells, you know, different subsets of conventional dendritic cells. There is much research which has been done with type 1 conventional dendritic cells, but we think that there could be a role also for type 2 conventional dendritic cells modulating Th17, and through that pathway controlling T-cell exhaustion and responses to anti-PD-1 therapies. And of course, there is much to do also in the field of preventing immune-related adverse effects, autoimmunity-like reactions, and how to combine these immune checkpoint blockers. As I commented in the presentation, timing could be one of the key issues, but well, we need to understand, continue understanding the mechanisms to translate these rational protocols to the clinics.
Sure. Yeah, thank you very much for your answer. And next question is from Professor Rodney. It says, do you see a future of frameshift peptide engineered T cells therapy for Lynch syndrome? You are muted. Please unmute yourself. Thank you.
Sorry, it's okay. Possible that T-cell therapy may come forward, but I think what's going to happen before that is vaccination therapy, where particular vaccines are going to be developed from frameshift peptides to then inoculate patients with Lynch syndrome, which should theoretically eradicate any tumors that begin to develop, especially if you can find frameshift peptides that are common between a significant number of individuals with Lynch syndrome.
Okay, sure, thank you. The next question is from Dr. Marcelo again, I think. So it says, it has been observed that anti-PD-L1 therapy includes the high level of INFG, which is potent inducer of IDO. Do you find any correlation of inflammasomes with IDO expression in tumor microenvironment, or has any plan in future?
Yes, we have done some experiments related to this potential relationship between inflammasomes and IDO. So what we have seen is that when we block the inflammasome inhibitor with a compound, we observe an increase in IDO activity and expression in the tumor microenvironment. The same result was observed with the TMEM176B knockout, which enhances also inflammasome activation. And this unleashed inflammasome activation is associated with increased infiltration of the tumor by CD8 T cells expressing interferon gamma. So if we delete CD8 T cells, we can control IDO expression. And this IDO expression is mediated by tumoral cells. So the idea is that when we unleash inflammasome activation, we are recruiting CD8 T cells, but these T cells are triggering IDO expression in malignant cells. So far we did not succeed in improving the antitumoral effect of our compound by concomitant inhibition of IDO. So we performed the double therapy, IDO inhibitors and inflammasome unleashing therapy, and we were not able to see an improved antitumoral effect, but we are working on the protocol trying to increase the dose of IDO inhibitor, because as I said before, we do see that we are triggering IDO activity and expression when we unleash inflammasome activation.
Okay, thank you very much. Last question is from Professor York. This is about the methyl marker identification by machine learning. So big data scientists or artificial intelligence people, they are talking about the treatment algorithms. They say that we can, define softwares or models which can give the treatment algorithms, and the scientists can look for the potential drugs that can actually bind to the protein structure, or the methylated or translated or post translational modified proteins. So do you think that this treatment algorithm offered by the big data scientists or artificial intelligence could be merged with the methyl marker identification in the cell population that you are mixing? Please unmute yourself.
Same problem.
Thank you. No problem. It's okay.
No, absolutely. I mean, I'm not an expert in machine learning myself. I'm a molecular biologist. So we are just applying it and collaborating with people who know about the algorithms better than we do. But yes, the idea is really, since from our experience, we get much better data, or much better results rather, by applying these algorithms, or there's a couple of them that we use so far extensively, and the results are just much superior to what we could achieve beforehand. So I'm pretty sure that mixing and merging this, the machine learning algorithms with a lot of data that we are producing and have been producing could make quite a difference, as a matter of fact. So without new data, we might be able to generate new data and new results, as a matter of fact.
Sure. Okay, thank you very much. With this, I end this session, and I'm very thankful to you all for your time and your participation and your presence here. And it's a great opportunity for us all to hear from you directly. And I hope to see you next time in person here when we have a symposium, not so virtual, and we'll discuss the ideas further. So thank you very much, all the audience and speakers. And I would like to end the session now. Thank you. Bye-bye.
Thank you.
Thank you very much.
Thank you. Goodbye.
Thank you. Bye. 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 Bye.